Sir, we are live now.
Great.
Jenny. Oh, we are live now. Okay.
Yeah, okay, Rohit. Yeah.
Hi, Mani. Good morning.
Good evening. Hi. We just kept Rick awake late, and then we are just going to keep him awake again today.
Terrific. Lovely to see you all too.
Hi, Mani. A very good morning. Yeah, good evening, guys. Yeah, good evening. Yeah. So I guess
Mani, we have Dr. Andreas Moller. He has also joined in. Good evening, Andreas.
hi, good
evening.
Good evening, Andreas.
So it's 7:02, and I think Prashant, I think by 7:05 we should
Start.
yeah.
Okay.
So, Andy, I'm not sure I'm going to make it to the end tonight, so apologies in advance. It's mostly about what I have to do with. It's about what I have to do in the morning. I'm sorry that otherwise I wouldn't have a problem, but I don't have so much flexibility. Anyway, I'll see how I go, Andy. See how I go.
It's Sunday morning tomorrow. Come on.
Yeah.
Two nights.
Special day.
Special
day for me.
You can make it today. Sunday. He goes for a sailing. Rick goes for a sailing almost, I guess, every weekend, right, Rick?
You know, I have to go sailing in the morning. Yes.
Yeah. That's
right. Not every weekend, but tomorrow it's the fixture day. So it's good for everybody.
That's good. I guess. I hope you have a warm water.
Oh yeah, it's really fine. Been colder than usual this year, and tomorrow's very nice weather.
Want me to come along?
I know you're going soon too, Andy. I hope so, right?
Just waiting. Yeah.
Yeah. Tomorrow we have just our usual thing.
Hi, Dr. Kasi. Thank you for joining in.
Hi, how you doing?
Welcome.
Good morning to you, Dr. Kashi.
Hi, how you doing?
Good.
Prashant and Dr. Kasi meet here. So Prashant has organized this and put together the meeting, and me and Mani we are standing support and in every which way. And so he's getting to meet you right now.
So,
Oh, that's wonderful. Yeah.
so good morning, Dr. Kasi, and welcome. Thank you very much.
Likewise. No, thanks for having us. I think it's. I was looking at the 2 days of talks. It's a very comprehensive effort.
Thank you very much. Appreciate.
Thank you for accepting.
Hi, Prashant.
Hi.
Good evening. How are you?
Good. Good.
Good, good, good.
Good evening, sir.
Hi. Good evening, Samanthi. How are you? Good to see you. Always smiling. You look full of energy all the time. That's nice.
Thank you. Thank you. We all, I think all of us slept late, and Rick the latest.
Yeah.
And he's still smiling. We are
Good, good.
So,
Yeah.
Thank you, Dr. Govind Babu.
Nothing at all. I learned so much yesterday.
Yes, all of us did, and just so many different views, discussions, and
Absolutely, absolutely.
amazing.
Yeah. We are just actually starting our molecular tumor board at HCG soon.
Oh,
I
excellent.
think that's the call of the time.
Yeah, we've just started a 500 gene panel recently.
Yes, the strand advantage.
Yeah.
Yeah, I've already done some.
Yeah, same here.
RNA, yep. Nice.
I think we're finishing 100 patients soon, then we'll see the data.
Fantastic.
Hi, good morning, Klaus.
Good morning.
Thank you for joining us. Thanks a lot.
What is the time now in India, just to be sure?
It's 7:08 p.m. And
7:08. Okay. Well, it's afternoon here. It's 2:37 p.m. afternoon here. A beautiful sunny day with cold snow and winter time here.
It's a very good afternoon to you.
Same to you.
So,
Hi, class.
Yeah, please. Mani, I think Klaus didn't hear you.
No, hi, class. I said hi.
Hey, Mani, good to see you. Great.
Good to see you as well.
So I think we are live, and Prashant will be starting the meeting in a minute.
Yeah, I think 10. I think 7:10 should I start?
Yes, please. So we have Klaus, we have Rick, we have Andreas, we have Dr. Govind Babu, We have Dr. Pashtun Kasi, of course, Mani, myself, Prashant, and our respective teams. We've all of us logged in, and I think we're all set to go. 30 seconds.
Cool.
Hi, Klaus. Good to see you again.
Good to see you, Rick. Looking forward to your talk.
Thank you. Thank you very much. It's great to get together with everybody like this, actually.
Yes,
Really
that's
nice.
true.
Even if it is virtual, yes.
Yeah. I really got a great kick out of it.
We have to find out how to make a virtual beer drinking or wine tasting. Huh? I mean,
virtual.
Oh my goodness. Okay, so let's start. Seventy. Hello.
Yes.
Yeah, go ahead, Prashant.
Hi. So hello, everyone. Good evening, good morning, and good afternoon, depending on which part of the world you are connecting. So we are starting our second day, and you know the theme of the meeting, liquid biopsy, a game changer. And something I really liked what Dr. Mani Sandarai said yesterday, that everybody who is associated with the meeting and all the speakers, they are actually the game changer. And the theme is really the right way to start. The COVID time, I would say that what we were just discussing, that brought us all on the virtual platform. But what I realized really, that many of the time when we go in a big meeting and sitting in a bigger seminar hall with multiple people and many people around, what I realized yesterday that somehow with the virtual platform bringing us more closer, you can see your speaker very closely. And that gives us a belonging, and which I was realizing yesterday. Yes, we are very open to interact. And the theme, what I mentioned earlier, that the theme of the meeting is that we are coming on the same platform where we can look forward for the collaboration. We will look forward for the discussion. And yesterday we realized that many of the time we couldn't able to take the questions. So I would encourage the participants, please don't get disheartened. Please put your questions, and I would request the speakers, whenever they get time, they will answer your queries. So let's start with just an introductory slide. The liquid biopsy, I think this is something what we call a game changer, but the cancer, the way we see and the way clinicians perceive the cancer and the way they monitor, clinically, it is based on the imaging. And the imaging tells us that, okay, the tumor is growing if the size is getting bigger or it's getting shrink. If you see the lab which are working, molecular pathology, so we use different kind of technology to understand, again, the tumor progression or the biology of the tumor. And we do use many different technology in the lab. And one of the very popular one is immunohistochemistry. And that is where the pathologist use immunohistochemistry to see the tumor, to validate the tumor. And there are other techniques, including the FISH and the FACS and many more. But what I would say that something which make a revolution in the genomics, and not only the cancer, but in I think many more diseased condition, is the NGS platform. And I would say that last 1 decade, if you see, the NGS made a huge difference. And the cost, of course, the costing decreased a lot over the time. But what NGS facilitated us, that we could actually decode our genome. We could understand the kind of mutations happening over the time in the diseased condition. And this is where the NGS, you could see that now the clinicians are using it routinely. And either you would like to find the information about the whole exome sequencing, whole genome sequencing, or kind of somatic mutations, it's indels, structure variations, translocations. So it is actually giving us in detail understanding of the genome. However, when we are talking about the liquid biopsy, and this is the time, I would say that maybe again, my emphasis would be last 5 years, I would say that I could see it is really, really coming up, and clinicians would be the worthy people who could tell you more that how routinely they are using it and what advantage it is giving. And this is something which is really advantageous using liquid biopsy, is that it is in the real time. So you can ask the patient multiple sample, which may not be possible when you use the tissue biopsy. So liquid biopsy is something that we call it non-invasive. I would call it minimal invasive. And when you call minimal invasive, you could collect the blood and you could ask the similar kind of question, which you could perhaps get it using the tissue biopsy. So this is where the liquid biopsy is bringing us a huge opportunity to understand the follow of the patient, understand the relapse condition. And many of the time when patients are not in a good condition, you may not want patient may not want to go through the surgery. I think liquid biopsy is the way to go forward. So I would just conclude the introductory slide of mine. And I'm very, very happy to introduce the first speaker today, Professor Rick Thompson. Rick is a great friend, colleague, mentor. And I have been associated with Rick for the last 4 years. And the way I learned the way he does science, the way he has a passion for the science, it is extraordinary. So just to brief you about the Rick achievements, Rick is professor, translational cancer biologist. He has done his PhD from Griffith University, Brisbane, Australia. He undertook postdoctoral from NIH. And he is appointed as a professor in breast cancer research and a leader of the chronic disease and aging team in IHBI in QUT, which is one of the premier institutions in Australia. In 2016, Rick took up the role of associate director of IHBI at the Translational Research Institute. I would say that he's very active in the Society of Science, and I would say that I have seen it very closely because we somehow managed to get a grant, which was called Indo-Australia grant. And under that grant, we were actually supposed to do some awareness program. And he traveled to Bangalore a couple of times, and he was part of the awareness plan and the program for the breast cancer. Rick is the founding president of AMATA in 2004. And the biggest contribution I would call from the Rick is he has co-founded the EMT Association, which many of us are very closely associated. It is called TMTIA, in 2003. And he had been elected as a president in 2013. And we call it that this particular association, which actually brought all leaders who works in the area of EMT, including Dr. Sundaray Mani. And they are the innovator and they are the leaders who really translated the EMT discoveries, how we could understand EMT better in diseased condition. Professor Thompson has 2 major programs of research in breast cancer. One is mammographic density and the other one is epithelial mesenchymal plasticity in CTCs. So again, I'm very pleased and delighted to welcome Dr. Rick Thompson. And we look forward to listen his talk. He's going to talk about the human-specific analysis of CTCs in breast and prostate cancer xenograft, insight into the EMP hybrid state. Thank you, Rick, for joining us.
Well, thank you so much, Prashant, for that very kind introduction, and to you, Suwanti and Mani, for the wonderful opportunity to participate in the meeting. And I'm just switching. Can you see my presentation okay?
Yes, absolutely fine.
Yeah, thank you. Thanks for a really kind introduction and reminding me of the wonderful visits to Bangalore, including to be there with Mani at the time of the Indo-Australian EMT meeting that you helped us host, and also to visit Suwanti in her hospital in Mumbai on the way. So it was really very nice. So thank you for that. So really enjoyed so much the program yesterday, and a terrific update in so many areas, particularly in the clinical area. And we take a little bit of step back now to the translational side in my talk at least, and look at PDX models of prostate cancer to see what we can learn about circulating tumor cells in this little bit more, model where we can have a little bit more reproducibility between the subjects and really hone in on some of the biology and potential opportunities for better utilization of CTCs. Just a quick look at where we are. This is where I am right now. In fact, I'm just off to the screen at the left. This is Brisbane. If for many of you, I think that maybe haven't been here, and also we hope that you will come soon. This is where we work in the Translational Research Institute. It's a multi-institutional building, and it's associated with the Princess Alexandra Hospital here. Brisbane City, you can see it in the distance. Andy Moller, who'll talk later, and he's here, is working just on the other side of the city. And you can see the river snaking around here. Just at the back, you can see the sea and the barrier islands, and we're sitting on Moreton Bay. So it's not the most glamorous photo, and there's a lot of work going on in this area at the moment. We're having a new rail system put in. But this opportunity to work in close proximity with the hospital is great, and also with collaborators from different institutions. So as Prashant kindly mentioned, I have a long-standing interest in epithelial mesenchymal transition and epithelial mesenchymal plasticity. And just to remind you, many of you I think have seen this, and Jean-Paul is really the one who has introduced this concept in the very early days of EMT being used by cancer cells to be able to move away from the carcinoma, to lose that connectivity with the epithelial cells as they become mesenchymal-like. You'll find also some hybrid cells denoted orange here. And the mesenchymal cells are able to degrade through the tissues and migrate and get into the vasculature. And this, of course, is what we're mostly interested in here today. And the talk is about circulating tumor cells, trying to understand what we see in the bloodstream in these PDX models of prostate cancer. And we're building on some work we had done earlier using human-specific RT-PCR approaches to look at the transcript levels of EMT-associated genes and other genes of clinical and context significance in the circulating tumor cells compared to the primary tumor. In the breast cancer, we could compare that to metastases, such as the bone metastasis here, local recurrences, or in the case of the breast cancer lung model we used for lung metastasis. There's opportunities to target EMT and epithelial mesenchymal plasticity. And number one, in targeting the mesenchymal phase. Number two, in targeting these hybrid states, which we have becoming much more aware of, and there's much more focus on these hybrid states. And indeed, it's been shown by Cedric Blanplain and Yevgenka Pastuchenko how important the hybrid state are, that they have a stronger metastatic potential than the mesenchymal state. I've got this thing in front of my thing. And so when you get to the metastatic site, or even the local site, it's very clear that these metastases are epithelial compared to the mesenchymal precursors that you might see in the bloodstream or in the bone marrow. And certainly in the case of the MDA468 model, which we did study and have published, I'll show that paper a little later, we could see that the signals that were changed in CTCs, mesenchymal ones, and some epithelial ones also were increased in the CTCs compared to the tumor. They were returned to the tumor levels in the metastases. So there's certainly this is consistent with a lot of evidence, and the idea that cells may need to undergo a mesenchymal epithelial transition at the secondary site. However, it's been difficult to clearly demonstrate that transition. And it's a mixture of cells that are becoming mesenchymal and then becoming epithelial again, and the possibility that some cells are actually transgressing from the primary tumor in the epithelial state. Whatever it is, they need to be epithelial to colonize those target sites. So I just wanted to introduce briefly prostatic cancer. The study is a wonderful collaboration with my wife, Elizabeth Williams, who's been studying prostate cancer for many years. I'm less familiar with it. But I think many of you will understand, and what we want to do is have models which are relevant to the disease progression in patients. When prostate cancer appears, it's generally managed initially by surgery and radiation. It's often monitored and sometimes diagnosed through the secreted tumor marker PSA, or we also use the term the KLK3, kallikrein-like related peptide 3. So when you do, initially the patients respond to the surgery and radiation with a reduction in the circulating levels of PSA, and you have this period of successful treatment, and then you may see biochemical failure at some stage after that, maybe in 30% or so of the patients, which is then treated with androgen ablation. So almost all prostate cancer is AR positive, androgen receptor positive, and responds initially in most cases to androgen ablation. The increasing number of more effective agents and strategies for androgen ablation. And then unfortunately, in a small proportion, maybe 25% or less of the patients, you'll have a second PSA failure as the tumor cells become less dependent on androgen ablation. And this is termed castrate-resistant prostate cancer, in which case they're retreated with chemotherapy palliation. And so in order to model this disease, we have a number of patient-derived xenograft models which represent and show those different treatment responses. So what about CTCs in prostate cancer? Actually, there's quite a lot already done. They're very well described, and in fact, one of the best papers is by Klaus, who's here in the audience, his lovely review in 2019. It's a very clear prognostic value of CTCs when they're present, such that the number of CTCs seen in the prostate cancer at diagnosis associates with their overall survival. And there was a very nice paper just in the last week in Clinical Cancer Research indicated here, showing in metastatic castrate-dependent prostate cancer that the CTC burden at outset was highly prognostic in terms of the overall survival response. And so these patients had metastasis at outset. It's also been shown to be important and effective in monitoring disease state after therapy. So the change in the CTC numbers is indicative of therapy and clinical showing the clinical utility. At the liquid biopsy level, there's been also, we heard about a number of tests which are now approved for use in monitoring disease state and therapy potential in several cancers, and this includes prostate cancer with ARV7 having prognostic value and can be detected in circulating tumor DNA and is FDA approved for assessment in prostate cancer. And Andy will talk later, I'm sure, about extracellular vesicles, exosomes, and this has also been reported to have predictive value in prostate cancer. And probably more from my point of view also, there's quite a lot of work around understanding the biology of metastasis in studying CTCs and the genomic changes in circulating tumor DNA. So as I mentioned, we're using the PDX models, and these have been established and maintained by Elizabeth. So sorry, not all established by Elizabeth, but the BM18 model she established in the laboratory here in Australia. And we're very grateful to Eva Corey and Bob Vasella, who have provided a number of their LUCAP PDX models. All of these models must be maintained in mice. All of these models are androgen dependent, and you can see as an example the BM18 model in the blue. If you grow these in the mice without castration, they grow exponentially. With castration, you see the tumors disappear. In some cases, not so much in BM18, they're very androgen dependent, but in some of these models, example being LUCAP35 here, they respond to castration with a decline in tumor burden, and after some time, they do what's seen in people with a castrate-independent response. So we've used these xenograft models to try to look for initially CTCs and then characterize these CTCs by immunocytochemistry and RT-PCR using human-specific probes. I can't get, okay. So initially we actually did RT-PCR to select the xenografts we would study, but I'd just like to show you some of the immunocytochemistry data that we obtained with some of the models. Out of the 7 models, we ended up pursuing 4 of them because we could see reasonable evidence by RT-PCR of the tumor cells being present in the blood. And so here you can see by immunostaining some examples with keratin staining, cytokeratin 8, 8, and 19 as a mixture of antibodies and also Vimentin. And as has been shown by many, and Shyamala will talk about some wonderful work, and Min Yu showed beautifully in a science paper several years ago, being able to identify and stain these circulating tumor cells with cocktails of RNA probes for epithelial and mesenchymal markers. Here, using the Vimentin antibody, which is human-specific, we can have 100% confidence that this is a PDX-derived CTC. This one doesn't actually stain CK. This is primarily mesenchymal, Vimentin being a mesenchymal marker. But here you have a CK-positive tumor cell around about the same size. So these are larger sizes, much larger than the circulating mouse leukocytes, which you can see the DAPI stained nuclei here in the lowest panel versus the tumor nuclei stained here. And so there's a significant size difference in the cells which we're looking at here. These are small clusters here then, that's staining both Vimentin and cytokeratin, which we see to a variable extent in the different models. And we can see this cluster here with a primarily cytokeratin staining and no Vimentin positivity. And when we summarize the different models, as I said, we've looked at 4 of the xenografts. We see some very interesting results in terms of the heterogeneity, even within the same experiment, different mice. So these are different mice for the BM18 model. The total number of CTCs we could see by Vimentin keratin staining ranging from 4 to 400 in that model, or here 5 to 1294 in these 3 different mice. And then the LUCAP 105 having rather lower numbers, but still we could monitor these and we could study their gene expression by RNA. Again, too, the number of clusters varies a lot, but there's a clear relationship between the number of CTCs circulating and the number of clusters that we would see that are staining with either Vimentin or keratin or both. And we're very, very interested in this hybrid phenotype, the cells which are expressing both mesenchymal and epithelial markers here, Vimentin and cytokeratin. And you can see that the number of cells which are positive for both varies quite a lot as well. So here we're ranging between 0 and 75%, 0 up to 100% in some of the mice in this model. So we're rather surprised that it's so variable when we look at it in the by immunocytochemistry. When we did our study in breast cancer, we found that the gene expression levels were pretty tight when we're looking at the expression profile within the CTCs. But the number of CTCs that are existing in the different states is quite variable. We did notice some small CTCs, and these are interesting because size is often used as one of the key factors for physical isolation of cells by methods which are not using epithelial specific capture. And so not all of this is the large size cells that I was just showing you, but here's some examples of small keratin positive or Vimentin positive circulating cells. We're not, the keratin antibody, we don't know that this is human specific. We haven't actually tested that yet, this mixture of 8, 8, and 19 anti-keratin antibodies. And so we can't be confident that these are really CTCs. But certainly with Vimentin staining, we can see, and certainly there's a great concordance between keratin staining in the larger cells, but whether or not all of these keratin entities are CTCs. And so when we look for these small, out of the 3 mice, we could see small CTCs in 2 of the 3 mice or both 105 models. And so we think this is something worth pursuing to understand a little more about what are the actual numbers of these and what's their biological potential. So switching to the gene expression side, which is really where we started, you can see that the 7 different models that I mentioned earlier, when we screened these by just looking for the abundant housekeeper L32, we chose 4 models which had reasonable numbers. And here you could see we excluded the 141, the 3-5, and the 2-3 models because they only had 1 out of 7, 1 out of 9, or 0 out of 3 mice with detectable signal. But for those which we could, for those models, we then went out along and we were simply obtaining mice from the colony as it was being propagated by serial transmission of the PDX. And you can see we were able to accrue numbers that we determined should give us results based on what we've seen in breast cancer, using a 42 gene panel of human specific primers for EMT related and other contextual genes. And here you can see that the kind of a screening we did, actually we did PSA as well because it's quite abundant, or KLK3. And so you can see a plot here of the KLK3 CT. So the smaller numbers indicate higher expression, and the L32. And so as we go along this axis, we're looking at lower levels of expression. And you can see once the L32 is dropping here, we're losing the PSA signal. And so quite a few of the gene markers which we studied, we're not able to really detect because we dropped below the threshold of detection. And for those mice, we actually have to exclude that data because we're not certain what the level is. So this is a plot that shows you the number of genes detected out of the 42. It's 43 with the housekeeper gene, compared to the level of L32 housekeeper. And so not surprisingly, as the level of housekeeper drops as we go to the right, the number of genes detected drops also. And we set up a threshold so that we included any sample where we could see at least 5 genes in our analysis. And we also saw, not surprisingly, that the tumor weight of the tumor that was resected, and we were resecting the tumor, we're weighing it and then extracting RNA to compare the RNA levels of these genes with the primary tumor, with the RNA in the CTCs. The total number of CTCs was increasing in most models when we had a higher tumor, but not all. And in fact, it wasn't really a significant relationship until we pooled all the data. You can see the p-values here don't drop below 0.05. And the overall R value eventually is around about 0.4. So sorry that this is a very busy looking slide. And unfortunately, it is a lot of data for these groups of mice in the 4 different PDX models. And this is a heat map that includes all of the tumors that are in the blue here and all of the CTC samples from all of the models. And the different models are shown in this color band at the very top to be the BM18 in black, LUCAP yellow, LUCAP 70 yellow, 96 blue, and brown is 105. And the genes here are actually listed down the right-hand side, and they're expanded so you can see them more easily. And they're coded as well. So the mesenchymal are coded in green, epithelial associated are coded in yellow, and other genes are coded in orange. Housekeeper genes are in the purple color. And so the other genes relate to, some of them which relate to, in fact, hormone receptors in breast cancer, because we developed this study for this panel for breast cancer, or they relate to hypoxia or cell adhesion and metabolism. And so you can see that amongst the CTCs, and these are hierarchically clustered in an unsupervised way in both the gene expression and also the sample. and there's one group here, one cluster here, if you like, where they've got reasonably high expression of these mesenchymal markers, as well as high expression of many of the epithelial markers. And so we feel this is a hybrid panel. And the other cluster is another cluster here, the tight cluster, where there's also quite high expression of these epithelial and mesenchymal markers. And then there's others where there's actually not very much expression of these mesenchymal markers and more expression of these epithelial markers. The CTC burden of these samples is actually listed here based on L32 calculation and an estimate of the number of CTCs per mil of the mouse blood. So you can see this hybrid panel that we have focused on has quite high CTC numbers. And these samples here, where there's a little bit of a reduced representation of expression, the numbers drop to be quite low. And this may be part of the reason that we can't see the signal for all of these genes. So this is a summary, and apologies again, but this I think gives you the broad view. And it's reference to the breast cancer study that we have previously done with the MDA-MB468 model, as well as the ED03, which is a lobular breast cancer PDX, where you can see if the CTC's levels were higher in the CTCs than compared to the primary tumor, it's a red arrow. And so we published this and we called this dysregulated EMT. So here you can see the mesenchymal associated genes as a group, starting with snail. If there's a hash before the name, this was the significance, but actually now in this case, we can see if there's an arrow here, this was significant to 0.05. In the breast cancer study, we also reported some lighter colored arrows where this was significant before, but not after correction for multiple testing. All of the prostate cancer work, and this has been done by Sarah Hassan, who I think is listening. So hi, Sarah, and thank you for all of your great work here. This is just finishing her PhD on this. And this is following up from Anthony Tashidas and Ann Lee, who have done the breast cancer work and have finished their PhDs earlier. And so here, all of these arrows are significant after multiple testing correction. So you can see that the mesenchymal cluster here, quite a lot of those genes are upregulated. And in some cases, such as SERPIN-E1, here, this is quite consistent across all of the models. You can see there's a reasonable amount of consistency across the models and between the two breast cancer models as well as the prostate PDX models. A very interesting observation, which I meant to toggle a little later, was PSA. So PSA was significantly reduced in expression in the CTCs in all 4 of the prostate models. Epithelial genes, and this may be due to it being an epithelial gene. You can see that some of the epithelial genes, and including CD24, which is in the cancer stem cell group here, are reduced in some of the models, but not all. But a lot of the epithelial genes, as I said earlier, are actually increased in these CTCs compared to the primary tumor as well. CDH1 is actually only reduced in one model. It was increased in both breast cancer models. And Grady had like 2, a major epithelial promoter, transcription factor is increased in some of these models. EpCAM, interestingly, is reduced in the 2 breast cancer models. And this is something that has been reported and discussed a lot in terms of the use of EpCAM. So this is the hypoxia cluster. And you can see this is something that's been reported in the past by Raghu Kalluri. And we know that hypoxia is an important driver of EMT in experimental systems and in vivo models. And you can see that these 2 downstream mediators of hypoxia and HIF-1 alpha message itself are upregulated in quite a few of these models. And also the metabolic state of CTCs has been discussed in studies. And PGC1A, which is a major factor in mitochondrial biogenesis, is upregulated in the CTCs in many models. And ILK is an important signaling molecule for tumor cells and their interactions with each other and with integrins and ECM. And in addition, as I mentioned, these rather breast cancer orientated hormone receptors, ER and PR, are upregulated even in some of these prostate models, even in models which are ER negative, such as the MDA468. It's traditionally ER negative, but it had upregulated. So we see a lot of change, and we see some patterns emerging, even though there's such a lot of heterogeneity. And one which we followed up is SERPINE1, just to look more closely at the protein level, where we knew we could get a good antibody which would work in immunocytochemistry, and also PSA. And these are human-specific antibodies for SERPINE1, and it's a human-specific protein. It's not produced in mouse as well, so we can monitor PSA and SERPINE1. And so looking just quickly at the protein levels of the SERPINE1, and this is PI1, plasminogen activator inhibitor, and you can see some of these large CTC cells which are positive for SERPINE1 but not for keratin, as this one, or with keratin and not for SERPINE1, as we saw also in the keratin Vimentin analysis, and also co-staining for SERPINE1 and keratin in the LUCAP96. And here you see a cluster which is stained for both SERPINE1 and keratin1, and you can see these cells which are positive for both markers in a cluster here. And we did tend to see that there was an epithelial coating around these clusters. And so this is the summary data. And here you can see more cells. So, but the trends towards the number of CTCs in the different models with SERPINE1 keratin staining was parallel the cytokeratin Vimentin. So more cells are in these are the same mouse bloods that we've used for the keratin and the Vimentin. And again, though, you can see quite a lot of heterogeneity in the 3 mice in terms of the total number of cells detected and the proportions which are hybrid or positive for individual markers. And again, you can see that clusters are more prominent when you have higher numbers of CTCs. So there's a certain proportion of the cells which are clusters. And if you have more cells, you can see more clusters. So that makes sense. And clusters, as I'm sure many of you know, have been implicated by Dan Haber's group and Nicola Acito in having a more prominent metastatic potential. And then finally looking at SERPINE1 or KLK and KLK3 PSA. So again, PSA being a human stain, SERPINE1 PI1 human antibody, and we can see in the LUCAP96 2 examples of either co-staining or PI1 only, and in the 70 model PSA only or co-staining. And again, similar types of numbers and similar relationships and a similar indication of heterogeneity in these mice. So just in terms of a summary, I think the models are really great, and the approaches where we can study these CTCs in confidence that they're human, they're comprehensive staining. So when we do the immunocytochemistry, we're taking the mouse blood and we're cytospinning it. We're not isolating anything with any way. And the same with the RNA. We're taking all of the nucleated cells and we're extracting RNA and looking at that with human-specific primers. So I think we're quite surprised that that's so heterogeneous in terms of the models. And we didn't really have a chance in the breast cancer models with the cell line MDA468 to see if that was the same. And the EDO3 was another PDX. So to some extent, that may be intrinsic heterogeneity that's more likely to be present in a PDX than in a cell line xenograft. So it's an opportunity to look at what's representative of what's in the patient. So high heterogeneity in terms of CTC numbers, but also EMP status. And we're very keen to understand more the implications of the hybrid state, which is clearly present. The RNA analysis doesn't actually allow us to say, are there different cells contributing to those hybrid signatures? But the immunocytochemistry shows us that that's certainly the case. The clusters are more prevalent when there's more burden, but it has been proven that the clusters arise out of the tumor. And this is again by Nicola Aceto, originally from Dan's group. It's very interesting for us to notice this epithelial coating, that there's more epithelial-only cells around the periphery of the clusters. This is seen with the Vimentin cytokeratin staining. Unfortunately, there's no metastasis formed in these PDX models. None of them actually lead to overt metastases in the mice. So we're not really able to pursue metastasis. In the MDA468 model, as I mentioned earlier, we could see that there was a return to the primary tumor levels for most of the markers in the metastases. And as some of you will be aware, there's a certain amount of debate at the moment, and it's been difficult with lineage tracing experiments to prove that there's a cycle requiring EMT before MET, despite the fact that there's lots of evidence to support that. And the small size CTCs, which we've seen, may evade current enrichment methods, depending too on the marker enrichment, but some of them will. The EpCAM marker is very, very good. We had a wonderful presentation from Massimo Cristofanilli yesterday, who did the pioneering work with the cell search model in the breast cancer and continues to do great work. And studies which have really gone to a lot of trouble to isolate epithelial CTCs and non-EpCAM positive CTCs have shown that really the epithelial CTCs are the ones that have the predictive value. So they're certainly present, the EpCAM negative, and certainly the epithelial mesenchymal plasticity process is one which offers a lot of metastatic opportunity to the cancer cells. But when it comes to the prognostic value, it seems the epithelial cells are winning the race. Interesting to see, despite the heterogeneity that we saw, that there is very consistent gene expression changes across CTCs compared to primary tumors. It's interesting and surprising to see that PSA is strongly downregulated in the CTCs. Maybe others are aware of that, but I haven't seen that. And so PSA being a very important biomarker for prostate cancer, this is a very important finding. PI1 is a potential marker, and potentially PI1 and other of these markers which are upregulated in the CTCs may have some potential in molecular targets for therapies, but we need obviously to do more work of that. And there's consistency when we did the different types of stainings for the numbers of CTCs. So as I mentioned, Sarah has done this work for her PhD in collaboration with Elizabeth and myself, and we were also fortunate to be able to go together a couple of years ago to the Anadous meeting in Greece, her wonderful meetings. And so with that, I'd like to just acknowledge Tony Blick, who's also worked with us at TRI as part of this study. Saeed Alinez has helped us with the CTC staining. This was also facilitated by the core facilities at TRI and also the animal facilities at UQ. And some of the breast cancer work that this comes from Mark Waltham, Anthony Tacitus, Alex Dobrovic, and Ann Lee, Alex being in Melbourne, and the RNA analysis and other RNA analyses not shown, supported by Melissa Davis. And we have a long-term collaboration with Christine Gilles from Liège, who was working on CTCs and has also made a lot of great work in the MDA460 model. And I mentioned the contributions, which we're so grateful for Bob and Eva for those PDX models. I'm happy to answer any questions. Thank you.
Hi, Rick. Thank you very much. It was loaded with the data, and I could see that there are multiple questions asked by the attendees, and I have also a couple of questions. So I'll start with a question from Dr. Shyamala Maheshwaran. Her comment, great talk. And she had asked that, do you see AR receptor expression?
That's a great question. We know those models have AR, and unfortunately it wasn't in our panel, and we didn't think at the start, because we're breast cancer people, we didn't think at the start we must put that in. And in fact, the human specificity of those primers and all of the working and checking that has to be done means adding another gene to the panel is not so easy. So we haven't done that. And we haven't stained for AR either. But my expectation would be that it's there. And a lot of the time, even as those prostatic cancers progress to castrate resistance, the AR is retained, even though they may develop mutations. So it's an important thing that we haven't done, and it's a good thing for us to do. Thank you.
Okay, so Rick, there's another question. There's a query. Have you looked into the proteomics expression of the PDX-derived clusters, particularly in hybrid subtypes?
No, that we actually, and this will be quite difficult with the number of clusters being fairly low, only visible, and the overall, you know, those numbers, those are the numbers per mil of mouse blood, and you'll probably know there's not that many mils of mouse blood in the mouse. So sometimes we're really dealing with a very small number, and to isolate those clusters, we'd have to really work on that. But it's a great idea. I think it's most likely that we would need to do more staining the way we've done with candidates before we could get to do proteomics, but I wouldn't mind doing that.
Excellent. So there's another question from Mani. Is there a correlation between EMP CTCs and serum PSA levels?
Yes, and PSA levels are taken in these mice. There's not a correlation. It's really been done because we've extended this work to castration as well. And we're just accumulating enough data on that. And there we're monitoring and we're showing the PSA that goes down with castration. And we're also showing what's happening with the CTCs in relation to that. And it's surprising to see, we find quite a lot of CTCs, even when PSA levels have dropped and the tumor has pretty much disappeared, all but disappeared in relation to the castration, we still find CTCs. And we're now looking at the gene expression changes that's occurring in them. So that means really, Mani, that the answer is no, there's not a strong correlation. And it's very interesting that the message goes down for the PSA, but probably those CTCs aren't contributing, you know, so much to the total serum PSA compared to what the tumor would be making.
Okay, so Rick, there's a question from one of my PhD student, and I must need to take this. This is Bernali Dev, and she has asked that are there any reports of contrary level of gene expression of PSA and SERPIN from patient samples?
Sorry, contrary levels of PSA gene expression and what, sorry?
And SERPIN.
Oh, SERPIN. Yeah, we haven't really seen too much data around the SERPIN. And I don't think we've seen anything in relation to the any correlation with PSA, but I'd have to check that. Yeah.
Okay, so a couple of my questions, Rick. So just clusters you talked, and you know that I have a lot of interest with the CTC clusters. So I'm just curious to understand that what is something which decides a cluster in the blood? It's a patient. So it's a patient heterogeneity, I would say, or the cancer types, or maybe some other dimension could be. I don't know.
Well, all I can really say is if we did, you know, it's really great that we have that work from Nicola to show that you know, with marking of the cells that they come from mixture of cells in the primary tumor, because of that relationship and the possibilities that they're clustering, you know, experimentally. And then also you see a number of as we've seen also, a number of non-CTCs in those clusters. So really the observations are, if clusters are present, the prognosis is worse. And the work that Nicola had done in that, if the clusters, if you look at those experimentally, they have higher malignant potential, I guess on a cell per cell basis, that they have you know, already they've got some heterotypic interactions that probably make them stronger.
Okay.
I'm not sure it really answered your question, sorry.
So would you, because I just realized you have mentioned that clusters, more clusters, it denotes that high CTC burden. So do you mean that I would expect that in lung cancer, I would expect more clusters, or I would, compared to glioblastoma or any other, no, can I correlate with the mutation burden also, do you think?
They may. For us, it may be an experimental, in a sense, artifact. And if we see maybe 10% of our CTCs are clusters, if we have less than 10 CTCs in some of these, when we're looking at the protein level in some of these samples, we just may not be seeing it because we don't have a big enough field of view. So, and then it may also be tumor stream specific in the sense that what the, you know, I mean, breast cancer clusters have the JUP, the placoglobin. Other cancer types may not have that, and they may actually have their clusters constructed around different proteins. So I would say it's quite likely, but you know, it would need to be looked at.
So what we have done, we have actually made the quantitation of the clusters across 4 different cancers: lung, breast, esophagus, and bladder. And what we found that lung and breast, these 2 cancers, we always found that the diameter of the clusters were much bigger compared to esophagus and bladder. So we have done on multiple patients, and we found that it's showing the similar trend.
Yeah, so that really may relate to the cell adhesion molecules that are constitutive in that type of cell, or as one possibility, and other factors that will affect and influence, also microenvironmental cells that may or may not come out of the lung cancer or the esophageal and the bladder, but may be present, more prominent in the breast and prostate. So it's a really interesting observation, especially that the clusters have better culture potential, better malignant potential, and better prognostic power.
Okay, so I think there are many more other questions are there, and I would appreciate, Rick, if you could please answer some of them.
I will, thank you.
Chat box, and I thank you again for your time and excellent presentation.
Just one question, I think, actually a quick couple of questions. One is from Dr. Klaus Fantel. Did you see that question?
No.
Do you want to read it
Klaus? Klaus, oh, PSMA.
Yeah, it's just on the PSMA expression. Huh? Yeah.
Yes. So we haven't looked at that. We don't have it in our panel, but it's something we could potentially look at in the by immuno. Yeah.
And another question.
Another important one. Yeah. Thank you.
Regarding adhesion molecules in CTC clusters.
Well, I mentioned, I think, this placoglobin that has been reported by Nicola Cito and let me see, Govind, thank you for your question. Yeah, we haven't really looked. I mean, it's one of the things that we like to do more with this, with the immunocytochemistry, and that may be a way. But I think the questions and the ideas about working directly with isolated clusters, it's a great way. And if people have a know of ways to isolate those intact, I'd be very happy to hear from them. But thank you for the question, Govind.
Prashant, I think you should tell your clusters.
Yes.
Yeah, tell us.
Because this is what you have done, direct clusters, culturing them. Yeah.
Exactly. Yeah.
Great.
I think I'll get another opportunity to talk my
Sure.
talk and
Okay.
my work.
Okay. Okay.
Thank you.
We can
Thank
do
you
it in
very
the
much.
panel. Yeah.
Thank you.
Okay.
So, yes, Dr. Sevanthi, you can take the.
Thank you. Thank you, Prashant. And after that amazing talk and submission from 2 clinicians here on in the panel, a lot of the talk was heavy to absorb. Really, tried very hard to be able to get a lot of this, and wonderful, wonderful talk. Now we are going to move to the next speaker, and again, a huge honor to have Dr. Pashtun Qazi here today, this evening, and a submission that this was a request to Dr. Qazi to join the Star Power, and he very kindly agreed. And I've been following your work for quite some time on Twitter. Twitter brings the world closer, and it's a global world now. And I've really found the kind of work that you've done. I relate to that work, and in GI malignancies, this is something that I've tracked, followed in the recent times. And I feel that the field has moved towards really implementing the tests that we see the scientists doing in the labs and bringing it to the clinic. And that's how I came across the work that you do, and felt that this would be a value addition to this evening if you were able to come. So Dr. Kasi is the assistant professor at the College of University of Iowa, actually. And he did his internal medicine residency from University of Pittsburgh, and oncology fellowship from Mayo Clinic, and also postdoctoral masters in clinical and translational science from Mayo School of Graduate Medical Education. And as I said, he has, his niche is in ctDNA testing in gastrointestinal cancers. And I'm really inspired by his work. And one of the reasons why I reached out to him to present today. The main area where I have tracked the work is following early stage disease and adjuvant therapy allocation based on ctDNA, and also recurrence with monitoring ctDNA levels. So welcome, Dr. Kasi, to this wonderful collection of speakers, and it's an honor to have you. Please go ahead and take the floor. Thank you.
Thank you for having me. My email and our Twitter handle, as Sevanthi mentioned, is listed. We'll love to continue some of these discussion today online or later offline as well. These are my disclosures. I will be talking about off-label use of different commercially available liquid biopsy platforms. The key learning objective I wanted to focus on was the impact of liquid biopsies, primarily circulating tumor DNA testing here in patients with GI malignancies, focusing on the recent advances in the adjuvant setting as well. Just a couple of terms in terms of what we see in terms of in literature. You might see the term circulating cell-free DNA, cfDNA, cfDNA, because a lot of this technology originated in the world of prenatal diagnostics. And then recently, the same technology and some of the platforms are being used for tumors. You see the acronym ctDNA, and these are present in the plasma portion of the sample. As illustrated by Dr. Thompson's presentation earlier, the other aspect when we talk about the misnomer liquid biopsy, since there's no biopsy being performed, is the circulating tumor cells aspect. I'll briefly touch on some of the work that we're doing in our lab and some of the collaborations here on circulating tumor cells as well. But in terms of in the clinic, primarily a lot of this is pertains to circulating tumor DNA. And within ctDNA, the impact it's having in the journey of a patient with cancer is not just at diagnosis, but also the minimal residual disease detection, which will be one of the main focus of this talk, treatment response and acquired resistance in the metastatic setting. We'll be focusing on the latter three aspects of impact in GI cancers as an example. A couple of key landmark articles I wanted to highlight to the readers is this JCO publication, Journal of Clinical Oncology publication from March of 2018. This is actually the first time there was a panel in terms of looking at both the ASCO as well as the College of American Pathologists joint review, looking at this exponential growth of literature in the field of ctDNA in the clinic as to how it relates to the challenges, technicalities, impact, how to implement, how to interpret. Some of these are still relevant. And the key conclusion of this was that it wasn't necessarily ready for prime time. And I'll show in my talk that just within the last couple of years, the statement is already outdated. And then this recent other consensus statement just published in July of 2020 in Nature Reviews, again, is a NCI task force application. This was primarily focusing on colorectal cancer, but some of these concepts apply for other malignancies too, in terms of simple issues like, which tube to collect, how to interpret the data, the challenges in terms of other variables like chips. So these are two papers that I would highly encourage the readers and the audience to try to get their hands on them. These are available open access through some of the platforms. And how quickly has this literature sprung? 7 years ago, if you look at ASCO, there were only 12 to 13 articles that were talking about ctDNA, CTC, but now if you search the term ctDNA or liquid biopsies, just last year there were about 270 abstracts, presentations relating to diagnosis, specific aberrations, serial monitoring, concordance. I think we're kind of beyond the technicalities of the validation and concordance of some of these platforms. Some of these platforms are now FDA approved as well. In terms of, now these are not my slides, these are slides from discussants from our ASCO and some of the ESMO conferences. And like earlier joint statement said, it's not ready for prime time. I would agree with some of these discussants that at least for a metastatic setting and monitoring therapeutic resistance, it's already ready. And even for residual disease, I'll show you some interesting literature just within the last 6 months to a year that even this is being questioned and was a main topic of discussion at ASCO a week ago. Now, why is this relevant to GI cancers? In GI cancers, some of us already know that more young patients are dying of colon cancer, rectal cancer, and the age of colonoscopy screening has been moved to 45, but we see countless number of individuals in their 20s, 30s, and 40s who are getting diagnosed with colorectal cancer. And as you know, they are otherwise very healthy, very active, no comorbidities. We're still trying to understand why is that happening. And even within colorectal cancer, it's interesting, the sidedness matters. The patient's right side of the colon is very different than the left side, even though this is one long piece of tubing. The mutations that you find, the aberrations that you see, the biology, the survival is markedly different just between moving from the right side of the colon to the left. And interestingly, in the young onset colorectal cancer, it's more so rectum and descending colon, sigmoid colon cancers. And what that also means is there are more tools in our toolbox that can be used one after the other, from the traditional chemotherapies here on the left. We have all these biologics, salvage therapy drugs, immunotherapy, RAF and MEK inhibitors, and NTRK fusion inhibitors. And what that entails is, for patients with colorectal cancer, even the ones that we don't cure, patients are, this slide is, as you can see, this is more than a decade old. The improvement in survival from just a few months to several years, and I would say this number is now upwards of 40 plus months on some of the recent randomized trials published, which means that these patients are alive for years and years and years. And after exposure to multiple therapies, multiple mechanisms of resistance, multiple clinical trials, it is hard to imagine that the tumor that you're treating today is going to be the same, or to rely on a colonoscopy sample from 5 years ago to direct somebody's treatment today is something that from a research world, we know that these tumors evolve. There is temporal as well as intratumoral heterogeneity, but for the first time in clinic, we're seeing the utility of this in terms of liquid biopsies. My early explorations 5 years ago in liquid biopsy field was actually from a patient request, where this is again a similar 38-year-old young patient who went through multiple lines of chemotherapy, targeted therapy, and now had new liver metastases. And the question was, what else can we offer? And I suggested we should do another biopsy of this because, there could be new things in terms of HER2, other mechanism of resistance. And the patient outrightly refused because he's had multiple pumps and treatments and biopsies and surgeries over the year. He was open to anything else but an actual biopsy. So this was the first liquid biopsy that I ordered, and what I stumbled upon were very interesting findings. Illustrated here in different colors are different mutations present in the patient sample, and 53.8% is the variant allele fraction, with the denominator being the patient's own blood. So that's a lot of circulating tumor DNA. And colorectal cancers are notorious for very high shedding, which means that the samples are very reliable from a sensitivity perspective. But what's more intriguing to note is all these different other colors, green, red, these are all other acquired mechanisms of resistance that were not present at baseline, but were acquired. And in this particular instance, while we were waiting for some of these results, we did radiation to the liver metastases, and then we followed the patient. All these metastases, all these mechanisms of resistance actually went away, and then when the patient later had new lung metastases developed, when we repeated the liquid biopsy, interestingly, simplistically speaking, the clock was set back, and all the acquired mechanisms of resistance, EGFR, RAS, and other aberrations, they weren't present. So we were able to re-challenge the patient with the same drug that stopped working a year ago, and it started working again. Again, did we cure this patient? No, but were we meaningfully able to use another drug rather than just guessing what to recycle or reuse, which we do every day in clinic all the time? And simplistically speaking, the longer the time goes by from the last treatment, the better the odds of working. But here you can see the liquid biopsy can be used to direct recurrent recycling of the drugs based on presence or absence of acquired mechanism of resistance. Similar situation, another patient where the acquisition of NRAS happened, and that predated the progression on scans. Again, we were able to re-challenge with an anti-VEGF here, and then these clones can disappear. There's some work by us presented at ASCO a few years ago where the progression is predated by the mechanisms of acquired resistance on liquid biopsy, and then these clones disappear over time. They're not permanent. There is some elegant work from MD Anderson which looked at the half-life of some of these acquired RAS clones or EGFR clones, and it's interesting to note that it's about 3 to 7 months. And in our clinic too, arbitrarily, if somebody has had no progression in more than 6 months or 7 months or a year, that's what we kind of recycle. So these clones disappear over time, allowing for EGFR re-challenge to happen. And again, this is now something that's being talked about in different guidelines, ESMO papers, where there are two hypotheses. Maybe the maybe the resistant clone was there to begin with, and now blossomed on treatment while the sensitive clones went away. I would argue the opposite. I think these are truly acquired mechanisms of resistance, because now with these technologies getting more sensitive, we have had many patients who had tissue NGS as well as liquid NGS where there were no clones whatsoever, and they only appear at the time of progression or even before, when you do a targeted therapy treatment as illustrated here. As a replacement to tissue biopsy, this is multiple studies now are endorsing the use of CLIA-approved platforms for an alternative to tissue testing. Often for colorectal cancer, one problem is these are mucinous tumors, and sometimes tissue may not be sufficient. This is just one example of a patient of mine who actually went on the Beacon trial that led to the BRAF MEK inhibitor, the combination that's approved now as part of the guidelines with the anti-EGFR blockade. And in this patient, I had to biopsy this patient, several times to get tissue NGS confirmation because it was required for the trial to participate. But here, while I was trying to pursue tissue, and mainly I was pursuing tissue because I knew, as you can see illustrated here in blue, the patient clearly has a BRAF V600E mutation picked up on liquid at all the time points. So it's reliable, sensitive, and reproducible. And and it's encouraging to know that it's being now endorsed in guidelines and trials to be used as an alternative if tissue is not available. In terms of mutation burden, there's a lot of interest in IO and tumor mutation burden. A lot of the platforms are now reporting this. You see very hypermutated, colorful patterns in mismatch repair deficient versus proficient tumors. The other aspect that differentiates this from other tumor markers, the CEA or other glycoprotein markers, is the half-life is a lot shorter. So within hours to days, if you have an effective treatment, as early as 2 weeks, you could have prediction as to who's going to respond or not respond to treatment. I'll show you some trials that are incorporating this as a surrogate endpoint. Again, this is one example illustrating the previous slide. This is a patient of mine after 1 dose of Pembro. Within 3 weeks, you can see the amount of ctDNA went from 16% to nothing, and within the patient continues to have a complete response to immune checkpoint blockade for MSI high gastric cancer, who was on the verge of hospice and now is 3-4 years out. We also went on to publish this in the Journal of GI Oncology. Again, in patients where you do see response, you see the fall or clearance of ctDNA, and the patients who have persistence of ctDNA, or you could also check for MSI in plasma now, that's not a good sign. Again, these are a dozen different patients with MSI high, with ctDNA, and the clearance of ctDNA, as well as the clearance of MSI in plasma, correlates with response. And whenever you have presence of ctDNA, or for that matter, rise in ctDNA, or the lack of clearance of MSI high in plasma, these are patients who have progression later on scans. There's an editorial that we recently published highlighting this, as well as 2 other articles that looked at kinetics of liquid biopsies in predicting response to immunotherapy. Again, this is not just relevant to GI cancers, but would be applicable to anybody getting immunotherapy, which is the key therapeutic advancement in the last decade. And in terms of other beyond DNA, you also can pick up copy number amplification. HER2 is the most talked about target, not just for upper GI malignancies, but also colorectal cancers, breast cancer. Just a week ago, a new drug, trastuzumab deruxtecan, got approved for gastric adenocarcinoma, which is an antibody drug conjugate. You have tucatinib, other TKIs. So picking up these patients who are HER2 positive is of value. And also intriguingly for colorectal cancer, while the prevalence is about 2% in rectal cancers, if you expose these patients to anti-EGFR therapies, HER2 is an acquired mechanism of resistance. So you will not find these HER2 patients if you're only relying on the baseline sample. When you recheck samples at progression, that's when you'll find HER2. And some of these case series and reports talk about HER2 going up to 30 to 40% of the cases, which is a huge number compared to the baseline 2%. Problem is it's not safe, practical, or feasible, and patients are not going to be open to doing a tissue biopsy again and again and again. And that's where in a non-invasive, minimally invasive way, you could do liquid biopsy to pick up HER2. And as you can see from a sensitivity standpoint, this is data from one of the commercially available platforms. When they retrospectively look at the trial biobank samples, they were able to pick up the HER2 amplification in 46 out of 47 samples, equating to a sensitivity of 98%, correlating with response. A different type of aberration from mutation is fusions. While liquid biopsies are not the best tests, and they're not as sensitive as tissue NGS or RNA, for picking up fusions, when you do pick up fusions, it's very specific. So in these rare patients who have these fusion markers, the fusion inhibitors provide a brisk, dramatic, and quote-unquote curative responsive. So for the small minority who may have fusions, again, these can be present as an acquired mechanism. The liquid biopsy fills that clinical void as well. Switching gears to a different kind of cancer where liquid biopsy plays an immense impact on a day-to-day clinical practice is cholangiocarcinoma or any of the hepatopancreatic or biliary malignancies, including pancreas cancer, where tissue often is not sufficient for NGS testing. And this is a poster child for precision medicine. And interestingly, even though we treat the extrahepatic, intrahepatic, and gallbladder cancers with the same drugs, there are mutually exclusive, very different aberrations present. So for example, FGFR is pretty much only present in intrahepatic cholangio alongside IDH, whereas HER2, ERBB2 is mutually exclusive, primarily present in gallbladder and extrahepatic cancers. So while we treat all these cancers the same way, you could argue this is just simple plumbing with just bile draining from the intrahepatic to the extrahepatic with gallbladder and then going here. There are very different aberrations present in all these malignancies. And when present, when you do targeted therapy for these patients, this is just one example with many others in the report where these patients are alive years out. And in terms of drugs that are already approved, we have NTRK fusion agnostically approved, MSI approved. FGFR was the first targeted therapy approved for cholangiocarcinoma, present in about 15 to 20% of the cases. IDH, the data was presented at ASCO GI 2 weeks ago. It's now part of NCCN guidelines. This is also present in about 15 to 20% of intrahepatic cholangio. BRCA or DNA repair is 4 to 8%. HER2 is about 15 to 20%. BRAF, the data was presented in, again, Lancet, it's about 4 to 5%. MET is another couple of percent. So even if we just look at the upper sheet where you have available drugs or approvals, anywhere from 30 to 40% of the patients would have actionable aberrations, and it's not talked about as much. We talk about lung cancer and other cancers in terms of being a poster child for precision medicine, I would argue I don't think there's any other disease that has as many truly actionable aberrations, but often it's not necessarily the focus of topic, but liquid can fill the void because tissue is not sufficient a lot of times, because these are the patients who are diagnosed on endoscopic ultrasound biopsies, and the sample is never sufficient for NGS testing, let alone for diagnosis. And post-exposure to target therapies, again, there's a lot of heterogeneity that can happen. These patients can develop acquired mutations, gatekeeper mutations as mechanism of resistance. And again, similar to the other work that I showed you for colorectal cancer, these can be picked up months before the actual progression on scans. Switching gears to the other aspect, which is the most talked about topic in the last year, has been using it in the earlier stage setting in terms of, can we use ctDNA in patients with early stage cancer to pick up minimal residual disease, MRD, measurable residual disease, different terms being used, or can we use it as, does it correspond with outcomes and recurrence? And I'll show you some landmark articles, and they all have the same theme. When we talk about minimal residual disease, we're talking about the disease that may be present after a curative intent operation. So this is the patients in yellow who may still have residual disease, who may need mop-up chemo. And there are some patients who are already cured, but pretty much right now we don't have a way to figure out who are these patients who may already be cured after their surgery and may not need any toxic chemotherapy, or who are these patients who are not cured, where you may even want to intensify for sure, giving them adjuvant therapy. And as you can see, the imaging detection on some of these papers is anywhere from 8 to 12 months before the scan picks it up. The ctDNA can pick it up as well. And why colorectal cancer or some of these GI malignancies are poster child for some of these MRD assays is there's a lot of shedding in GI cancers as opposed to, for example, brain cancer on the right. The fact that you can even detect it in brain cancer, I think that's still remarkable. One of the landmark investigations is published in JAMA in May 2019. And pretty much you'll see the same trend. Every single patient who is ctDNA positive, the curve meets the x-axis. Every single patient has a recurrence. This is one sample post-op. And anyone and everyone who is ctDNA negative, it's not 100%, but pretty much 90-95% chance that 3 years, 4 years out, they do not have any recurrence. And then patients who have had serial examination, they can convert to negative after chemotherapy, and they are recurrence-free to date. And the ones who are positive and stay positive, or were negative transiently and turn positive again, all these patients have recurrence. So these series are small, but the patterns are pretty black and white. A different paper back in 2016, a different assay, curves are similar. Every single patient, ctDNA positive, recurrence within 2 years. Anyone who's ctDNA negative, again, 90-95% chance that you're cured. Different paper, 2008, Nature Medicine. Look at this. ctDNA positive, every single patient occurs within 2-3 years. ctDNA negative, recurrence free to date. This was an in-house panel from one of the universities that had published this paper from 2008. And this is another study from MGH. Again, look at the ctDNA positive. Every single time, there are no false positives here. If you are ctDNA positive, you will recur. And that's where the place where we want to make an impact. You know, can we use this more as a prognostic, or is it a predictive marker too, if we can convert some of these ctDNA positive to negatives? This is an assay from Roche. Again, these curves, you know, if I line them up with different assays, you may not be able to tell which curve is which, because every single time you see a very similar pattern. Compared to some of the traditional markers that everybody in the clinic across the world will be getting, whether it's here in the US or India or Australia, every other patient will be getting a CEA done for their diagnosis of colorectal cancer or other tumor markers. See how this compares to some of the ctDNA versus some of these protein markers in terms of stage 1, 2, 3, their ability to detect residual disease. One of the first hints that this may not be just be a prognostic marker was data from the IDEA France collaboration, where they did notice that if you were ctDNA positive, more chemo, i.e. here 6 months of chemo, it improved the recurrence-free survival. So some of the ongoing trials that I'll show you that we're a part of will try to answer this question, but I do think it's not just a prognostic marker, it's probably a predictive marker too. So in terms of summary and future directions, there's a lot of talk about, you know, circulating tumor cells and clusters. This is just work from our lab and our collaborations across the street here. This is from last week. A patient with esophageal cancer. As you can see clearly, This patient, just within 1 vial of blood, had 91 circulating tumor cells, and then immediately on chemotherapy, within 2 weeks, had the fall down to 0 and is responding to therapy as well. And in addition to the traditional stains to identify circulating tumor cells, we did HER2 staining, PD-L1 staining, and there's a lot of heterogeneity, like Dr. Thompson mentioned in his talk as well. Even though the HER2 tissue was negative, 50% of the circulating tumor cells in this particular case were positive for HER2 as well. So in terms of enumeration, beyond enumeration, there is an opportunity for in GI cancers to not just enumerate, find these clusters, stain them, and also we've done single cell sequencing. So you could use that for genetic analysis using panel-based testing where tissue may not be available. So it's not necessarily something that's in the lab. This is something that we're bringing to the clinic as well. In terms of, as you look at some of the data for ctDNA, you'll come across these new terms: tumor-informed, tumor-naive. Some assays require a piece of tissue to be sent before they can make a panel. Other assays are agnostic or naive, and they're combining epigenetic signatures. So when you read literature, see which assay you're dealing with, because the type of assay will also determine the sensitivity and specificity of, some assays may or may not be applicable to all cancers, whereas some may be relevant to only a particular kind of cancers. The way I see it, it's not something meant to replace tissue testing. It's not meant to replace clinical paradigms. Just like, if you see a patient in clinic today, you look at the T4, the nodal status, weigh in the MSI. This is one more tool. Even if you don't believe in the predictive power of the data, we decide on adjuvant chemotherapy today in clinic based on static prognostic variables. Here you have a dynamic, non-invasive marker which is showing predictive value or in terms of recurrence, outperforming all of these. Why not use it in clinic? In terms of metastatic setting, there are many trials. Colomate, this is using ctDNA. It's called the TAC-D study from MD Anderson. The Standard of Cancer Trial looking at target therapy for MRD positives. This is a study that we're a part of called the Bespoke study, which is using the tumor-informed Signatera assay, and we're trying to see if the intensification or de-intensification based on real-time results will make a difference in terms of adjuvant therapy or its ability to guide adjuvant therapy. There is the so-called COBRA study, which is using the Gardner's lunar assay in terms of stage 2A cancers, if they need chemotherapy or not. So there's a lot of activity going on. These are just a handful of trials with more to come. With that, I'll stop and would be happy to take questions and comments. And like I said, my contact is listed there, my email and our Twitter handle. Let me know if I can further engage in the discussion. Thank you again for having me.
That was a rocking talk, Dr. Kasi. that was amazing. And I, as a clinician, I learned so much. We have fans in the list here. Dr. Nirmal Raut has joined, and we were chatting on the side, saying what a great talk. So we have several questions. There is a few questions from the audience, which we brought on the chat box. So Rajat asked, do you do ctDNA testing before starting first-line therapy?
So again, not endorsed by the guidelines, but we have partnered with a lot of the commercially available platforms. As long as we can make sure that the patient will not end up with a bill and the copay is nothing more than anything meaningful, we try to do ctDNA and tissue testing at the same time before we start first-line therapy. Because gone are the days where you could just get away with giving FOLFOX with Bev to everybody for colorectal cancer, for example, and even for cholangio. The other reason why we do this testing is we have first-line trials available. We have a first-line trial for MSI-high colorectal cancer. We have a first-line trial for BRAF. We have a first-line trial for FGFR fusion. So, and a lot of these young onset colorectal cancer patients too, even knowing the status of BRAF, you would want to escalate chemotherapy to FOLFOXIRI with Bev in these scenarios, as opposed to treating them with doublet chemotherapy. So, yes, I personally try to do the liquid before starting first line. And the other reason why I do liquid, even if I can get the tissue, is there's some elegant trial from MD Anderson, as well as from there's a Nile study, and there's a study from Japan presented last year. There is at least, we underestimate the time we get the tissue NGS back. even in situations here, it takes me about 7 to 10 days to get a liquid biopsy result back, regardless of the platform. And it takes anywhere from minimum, I would say, 3 to 6 weeks to get the tissue NGS back. And it's not the NGS that's the limiting factor. If you talk to our pathology colleagues or pathology colleagues anywhere in the country, they'll tell you that, oh, they can get you the results back of any NGS testing within 7 to 10 days. That's not the limiting factor. As a clinician seeing a patient in clinic, that patient's sample may be sitting in a community oncology GI practice where the colonoscopy was done. So the number of hands that paperwork and other stuff that have to cross for sending the actual sample to wherever for analysis, that can take anywhere from 4 to 6 weeks. And the worst scenario is when they tell you in 4 to 6 weeks that the tissue wasn't sufficient. Well, you've lost all this time. So the rapidity with which you can get the liquid biopsy result back is also a key variable, especially if you're talking about anything first line in terms of incorporating the results.
I agree with you 200%, and I do them in my clinical practice. For any advanced patient, I start off right before I offer therapy. Do you see HER2 mutations in colon cancer and cholangiocarcinoma? Dr. Shamla Maheshwaran asked this question. I guess she means in liquid biopsy. Do you get to see HER2 mutations?
Yep, so one of the slides, and this paper has also been published by Guardant Health, where so cholangiocarcinoma, HER2, so you can see, so the actionable finding are actually the amplification. So HER2 amplification is seen in a 15 to 20% of extrahepatic cholangio, and in some gallbladder series, it's up to like 30%. So, similar to breast cancer and upper GI cancers, gallbladder and extrahepatic cholangio is where you see the HER2. Intrahepatic, not so much. And then within colorectal cancer, if you look at all patients, it's about 2%. And similar to like the picture I was showing you, it's more so the left-sided and rectal cancers where you see HER2 at baseline. But post-EGFR, you see HER2 in about anywhere from 30 to 35% of the cases. Again, these are more left-sided cancers, colon and rectal cancers. So not a small number.
Not a small number at all. And definitely something that has drugs that can be used to target. And so we should certainly look, the message is loud and clear. 2 questions from Dr. Nirmal Rao. And he asked, any data to use ctDNA to decide which rectal cancer can wait and watch post-CTRT?
Yep,
And,
so so while we're on the first question, so we actually have a protocol that we're developing to help answer the same question, because I think the bigger value, not just for rectal cancer, but even for esophageal cancer or any cancer where surgery, right now, for example, for rectal cancer, the patients who have a complete path response is almost 25%. If you do induction chemo, maybe some of the series go up to like 30, 35%, but majority of these patients have leftover cancer, and that's why surgery is considered the standard of care. However, there are many patients who will refuse an ostomy. They would rather die than have an ostomy. There are also patients who may not be medically appropriate to get surgery. So watch and wait, is a strategy that's being employed for a lot of different cancers. Rectal cancer in particular is one where there's a lot of talk and discussions and registries. Right now, how do we decide who are the watch and wait patients? These are patients who are MRI negative, endoscopic negative, tumor markers negative. Again, not to replace the current clinical paradigm, the perfect scenario would be to add ctDNA to that as well, so that the ones who are positive, well, maybe those are the ones where you may want to reconsider about your watch and wait strategy, or watch them closer, so to speak, versus sparing surgery in the ones who are negative.
Okay, great. His next question is, what if the ctDNA is still positive even after 6 months of chemotherapy in stage 2-3 colorectal cancer?
So that's obviously a bad sign. And some experts will argue, is it then truly minimal residual disease, or this is your stage 4 patient who just hasn't declared himself or herself on the scan yet? Dr. Parikh from MGH has a Stand Up to Cancer trial right now. She's exactly answering this question. So patients who are ctDNA positive after 6 months of chemotherapy, If they are BRAF, they will get a BRAF inhibitor with EGFR, just like the Beacon study. If they are HER2, they will get a HER2 therapy. If they have MSI high, they will get immunotherapy. And then if you don't have any of these actionable markers, then they will get FOLFIRI. So bottom line is that I think outside of a trial, well, that is a patient that is very likely going to be metastatic in the next few months. ASCO GI last week, they saw that these patients, had radiographic metastases within months after the ctDNA was positive. So overall, a bad sign. And, outside of a trial, that is a patient that I would just watch closer.
Okay, excellent. One of the last questions, and just request you to address some of the questions in the chat box later, those that we've not been able to cover. Zarian asks, how often do we come across FGFR3 amplification in gastric cancer?
So in gastric and upper GI cancers, last week at ASCO, in terms of FGFR, IHC, and ctDNA, they presented data using this new drug called BEMA with FOLFOX. And in that, the FGFR aberrations were present IHC-wise at least 30%. Now that was more so FGFR2. And for the first time, we have a drug that's targeting, it was a monoclonal antibody that they were combining with FOLFOX. In the ctDNA liquid, these patients had DNA amplification 2 to 4% of the time, but IHC expression in the tissue was about 30%. So again, not a small number. And for the first time, something that's non-HER2 that's being targeted in upper GI cancers. I am not directly familiar with the data for FGFR3, but the FGFR2 is about 30%.
Thank you. We have been sequencing upper GI malignancies here in India, and there's such varied alterations that we are seeing. It's an amazing discovery, mainly because this part of the world we may have different molecular signatures, and it's worthy to figure out what they are. I misspoke. I cannot not ask this question. Aarti asked, if there are multiple alterations seen on ctDNA, how do you decide which pathway to target first?
And that's where, it's a great question. That's where I think the liquid biopsies have to be interpreted in terms of clinical context. So if, for example, you have a patient with HER2 in colorectal cancer, and you also see KRAS, we know from our tissue other results that the presence of KRAS pretty much trumps the HER2, and these patients do not respond to HER2 blockade. So similarly, if you have any other actionable finding, for example, if recently had a patient with an NTRK fusion and MSI, some of them again are going to be disease context related. So cannot have a knee-jerk response to any actionable finding you find. Also have to be careful with the clonal hematopoiesis of indeterminate potential, especially in anybody above the age of 50, 60, 70. So for example, IDH is one, JAK2 is the other. Obviously, if you see this in a colorectal cancer patient, very likely it has nothing to do with colorectal cancer. These are present in the blood cells, so to speak. So again, you have to be very careful about interpreting any. You can't take the clinician's role out of the equation here. Some of the colleagues were talking about molecular tumor boards. I think these are great cases for discussion at molecular tumor board, where basic scientists, commercially available platforms, their scientific expertise, and clinicians together can then decide, what's the next best step or what's the next best target for these patients.
Excellent. I think you're rocking as a speaker. We're continuing to get so many questions. I can't ask them all. Just not possible. So I'm just going to read out a few comments.
I have a couple of questions.
Yes, Prashant, please.
Dr. Kasi, truly sensational talk. And I just received a message on WhatsApp from Dr. Prabhu from SCG Hospital, Bangalore. He's a GI surgeon, and he just asked the role of ctDNA in surveillance post-surgery in GI cancer to monitor recurrence.
And I'm, a co-PI for our biobank. My other PI actually is a surgical oncologist, and we actually are using pretty much, so we're part of the expanded access program with Signatera, Natera Oncology that has a tumor-informed assay. The nice thing about the tumor-informed assay is, it's not disease specific. They take a piece of the patient's cancer, they sequence it, They see what are the top 16 clonal mutations, and then they design a custom-built panel for the patient for that tumor in time. So they have data in breast cancer, prostate cancer. They have data in upper GI cancers. Pretty much for all our GI cancers, we've been using it. And last week at ASCO, the group from Denmark showed that the median time in terms of picking up recurrence was at least 8 months apart. The paper in JAMA a few years ago talked about 9 months. I've seen other data talking about 12 months. In my own experience, again, these were retrospective biobanks, so the clinician never knew that the ctDNA was positive, so maybe they weren't getting scans as often. In my own experience, more often than not, if somebody is ctDNA positive, that is a patient who already has a recurrence, if not detectable on the scan the same time. Usually 3 to 6 months later, I do pick it up on the scan. Recently, I had a patient who one of my surgeons operated, and there was nothing on PET scan, nothing on MRI, nothing on CT scans, MRI livers for a resected colorectal cancer, but was a very high risk for peritoneal recurrence. My surgeon actually went in with a diagnostic laparoscopy, and he saw, the PCI, the peritoneal cancer index of up to 15, and nothing on the scan. So again, did we cure that patient? I don't know, but we were able to start systemic therapy, and maybe we'll do cytoreductive surgery plus minus HIPEC, and we'll see. But I would imagine that if you pick up disease early, when it's minimal and not symptomatic, again, this is still a debate for ongoing trials. It's still better than dealing with the patient and being blindsided and now having gross, recurrences or visceral crisis where, quote unquote, curative intent treatment is completely out of the equation.
Just the second query, any change in mutations in ctDNA from primary cancer and recurrent cancers?
Not necessarily from a clinical context. Now, from a research perspective, yes, primary tumors, metastatic tumors do have different mutations. From a clinical perspective, unless you gave somebody a targeted therapy, which usually we don't do for adjuvant therapy, the mutations, the clonal aberrations, the APC, p53, KRAS, BRAF, they remain the same. As I showed some of the slides, in the metastatic setting, when you do anti-EGFR blockade, so not just colorectal cancer or cholangio or whatever cancer, the liquid biopsy serial testing or heterogeneity is more relevant when you expose the patient to a targeted therapy. You do not see variability in the assay, and I don't think the assays are helpful in the serial monitoring if you're just doing plain chemotherapy.
Thank you.
Thank you so much. And I think we'll end here. In the comment box, I see Dr. Rick Thompson saying really exciting clinical findings, great talk. Dr. Kazi and Nirmal says, thank you so much, Dr. Pashtun, amazing presentation. I'm a big fan of yours, your colorful tweets. So that goes without saying. And thank you again for gracing us and for joining us this evening. With this, I hand over the podium back to Prashant. And please go ahead with the next talk. Thank you. Thank you.
Yeah, the team, can you please put the introduction slide? So, hello. So I think the next speaker is Professor Klaus Pantel. And I would say that from the time of the inception of the meeting, we were very excited. And I think next 1 hour is going to be a treat to the listeners, because back to back, we have two stalwarts, which we are going to listen their work. And truly speaking, whatever I learned in liquid biopsy, and I have started working on the liquid biopsy almost 8 years back, when I was working with Professor John Paul Thierry in Singapore. And the very first thing he told me when I started working in this area, he just told me that, Prashant, go and read the paper from Klaus Pantel and Daniel Hammer and Dr. Mahestran. And that this is where I think I learned, reading their work, in-depth analysis of CTC, ctDNA. And I'm really, really honored to welcome Dr. Klaus Pantel and to introduce him to the attendees. So Professor Pantel, he is a chairman, Department of Tumor Biology, University Medical Center, Hamburg. Professor Pentel is known for his pioneer work in the area of cancer metastasis, micrometastasis, CTCs, ctDNA, microRNA, and he has published more than 350 publications in excellent high-ranking biomedical and scientific journals. Professor Pentel is coordinating many different kind of, I would say, European TransScan, CTC scan, EU IMI scan, and Cancer ID. So he is very actively involved in the European consortium, where they are collecting data across Europe on the CTC and biorepository work also. He serves on the editorial board of many international journals, and he has been awarded AACR Outstanding Investigator Award for the year 2010. He has also been awarded German Cancer Award 2010, ECR Advanced Investigator Award 2011, and many more awards which I cannot name. So it's really truly my privilege to welcome Dr. Pentel, and I'll pass this to Sundari Mani to please take and please introduce him from his side.
Thanks, Prashant. You did a great job introducing class, and class is a good friend, and as you said, he's a pioneer, and I'm really looking forward to listening to his exciting talk. Thank you, class, for making time available for this exciting symposium and providing your insights for us to learn even more. Thank you. Without further ado, I pass the podium to class. Thank you.
Great. Thank you very much, Mani, Prashant, and Sevanti for the invitation and for the wonderful introductory words. I could keep listening to you for another hour, but now it's my turn to work. And I really enjoyed this excellent meeting, and I hope I can contribute something in the next 20-25 minutes. So what I would like to talk today is about, of course, liquid biopsy and the path from the discovery to the clinical application. So as all of you know, that liquid biopsy allows a comprehensive assessment of blood-derived biomarkers. In the early stage, the tumor cells and the tumor cell products, such as the ctDNA, are derived from the primary tumor, or in a bit advanced stage from lymph node metastasis, and in even more advanced stages, the tumor cells and tumor cell products are derived from distant metastasis, for example, metastasis in the brain, metastasis in the liver, or in the bone marrow. So the assessment of these tumor cells and tumor cell products allows you a nice real-time assessment of tumor evolution in individual cancer patients. And together with Catherine Alix-Panherbier, 10 years ago, we came up with this term liquid biopsy, and we are very pleased that it has become so popular. So the two main analytes are circulating DNA. We heard a lot about circulating DNA already in the previous excellent talk. And of course, you can determine the tumor derivation of this DNA by looking at tumor-specific aberrations such as translocations, deletions, point mutations, chromosomal aberration, epigenetic marks, or amplifications. And in the cellular part of the blood, you can capture the circulating tumor cells, and Rick's talk was already excellent on the CTCs. And without going into any detail, there are actually 3 steps of the CTC analysis. The first step is the CTC enrichment. So we can use, for example, antibodies to EpCAM or other epithelial markers to enrich CTCs, like Massimo has shown for the cell search system yesterday, or we can negatively deplete the leukocytes, or we can use physical differences between CTCs and leukocytes to enrich them, for example, in increased size or deformability, reduction of the CTCs. And the second step, then we need to do a single cell identification. We can do that with immunocytological markers. Usually we use keratins for the epithelial tumors, or we can look at specific transcripts, or we can look at the excretion of tumor-specific proteins in the EpiSpot assay. And finally, we can do a downstream molecular characterization. And here we can characterize the CTCs at the genome level, at the proteome level, at the transcriptome level, and we can also do some functional assays. I will give you some examples in the next minutes. Regarding the ctDNA, I think we had a very nice network here in Europe with 40 institutions. It was called Cancer ID. I had the pleasure to coordinate that from the scientific part. And we just published a few papers on the pre-analytic and analytical conditions. And I just want to point out, it's very important to have very stable pre-analytic and robust analytic conditions, because otherwise you don't measure what's going on in the patient, you just measure variance of your blood, collection. So very important issue really, to have robust pre-analytic analytical conditions. Many of the ctDNA detection systems are, of course, dependent on NGS technology, next-gen sequencing. However, there are also some NGS-free detection systems. We have worked together with a company, for example, to develop a panel in melanoma with 13 genes, 65 mutations, including, for example, BRAF, which is relevant, of course, in melanoma patients. And the advantage here is that you have a high sensitivity down to 0.1% allele frequency, and you get this result without any bioinformatics needs within 3 to 4 days. What are the clinical applications actually for liquid biopsy tests? Well, it's of course early detection of cancer, which is, I think, very, very interesting, but also not so easy, I would say. And then there are the applications in patients that have been diagnosed with cancer. There is a response and follow-up, of course, to actually real-time monitor the response of a treatment. Then there is a determination of tumor evolution, and particularly the emergence of alterations associated with resistance to therapy. And of course, there is the goal of detecting minimal residual disease, which was also pointed out in the previous talk very nicely. And of course, this would also allow us eventually to start our treatments before full-blown metastasis signals incurability. So let me go through the different applications. Regarding early detection, we started a study called the Hamburg City Health Study 5 years ago. We have seen almost 45,000 Hamburgers between the age 45 and 74 who actually came to our university hospital. We have an own building dedicated to the study, and we collected a lot of biomaterials, for example, 120 ml of blood plasma, and of course, many data sets. And I think this is a very nice study for collaboration if anybody's interested in early cancer detection. We have actually in Europe 2 projects where I'm involved with my team. The first one is an EU Marie Curie network for PhD students. It's called the European Liquid Biopsy Academy, coordinated together with Tom Würdinger from Amsterdam. And here the focus is on early detection of lung cancer. The second EU project is called ProLipsy. Here the focus is detection of prostate cancer, and particularly clinically relevant prostate cancer. And I'm coordinating that, and the PIs are from different countries, including Dr. Lix-Panabir, Dr. Bonci, Dr. Budna, and Dr. Lianidou from Greece. The second application is, can we do a better risk prediction of patients and improve current tumor staging? So in this context, we found out that the CTC number or CTC count in a blood sample is tightly associated with an unfavorable prognosis. For example, in breast cancer, many large-scale studies were performed, but also in other types of solid tumors, such as bladder cancer, head and neck cancer, testicular germ cell tumors, colorectal cancer, pancreatic cancer, in skin tumors, we have such data. So in breast cancer, this has been now introduced in the AJCC Cancer Staging Manual as a new classification, CM0I+, which means no signs of distant metastasis by current imaging, but the presence of isolated cells in the blood or other tissues. And we can use this tool to enrich actually high-risk populations in clinical trials. Another question that we're asking in my new ERC grant, it's a second advanced investigator grant, there we ask the questions, do diagnostic biopsies or surgery or radiotherapy, so any kind of local perturbation of the primary tumor, does that lead to the mobilization of circulating tumor cells? And does it has any clinical relevance? And we are also analyzing the biology of the cells that are released in terms of viability and proliferation marker and so on. So this is a 5 years project that addresses this provocative question. The next application is definitely that we trace tumor evolution. And here we are, of course, very much interested in identifying minimal residual disease. And using ctDNA or CTCs, we are now able to identify minimal residual disease earlier before the imaging system signals really that there is full-blown metastasis. So we can use now these signals from the blood to identify patients with MRD, and that may lead to an earlier therapeutic intervention. So the vision here is not just to detect the subclinical disease and monitor it, but also use it for earlier intervention in the future trials. I mean, you have seen excellent data in colon cancer in the previous talk. Let me show you a few data on breast cancer. This was a study from Coombs and Shaw, published last year in Clinical Cancer Research on 49 patients. So a small study, but very nice findings. You see on the left side, the Kaplan-Meier plots that if the ctDNA had been detected during the follow-up period, the prognosis was dismal compared to those patients where ctDNA has been not detected, the black curve. And on the right side, you see different changes in ctDNA and as a tumor marker CA15-3. And you see really that the ctDNA had a quite significant lead time between 258 days and more than 700 days before the tumor marker imaging signaled that there was metastasis. So promising results, but don't forget 49 patients. These are the data on our CTC analysis. Here we detected CTCs 2 years after initial adjuvant chemotherapy. You see that we analyzed around 1,000 patients. And you could see here with the Kaplan-Meier curve, the red curve, it signals the prognosis of patients with CTCs and the blue ones without CTCs. And you see that the detection of CTC had a prognostic relevance. So with this, we can also now identify patients that have still MRD years after initial therapy. And the vision now is, and that was here actually suggested by Dan Hayes and me a few years ago, that this could give now the chance to do so-called post-adjuvant clinical trials. And we have just, we will see more and more of these trials in breast cancer, but also in other types of cancer in the future. The next application is liquid biopsy in metastatic patients. Of course, here we want to monitor the ctDNA and CTC counts to see whether the therapy works or not. And we also want to characterize the CTC and ctDNA to see what kind of therapy could work and do we really already see resistance to the initial therapy. So in terms of therapy targets and resistant mechanisms, there are two types of analysis. On the left side, it's the genomic aberrations that could indicate druggable mutations or resistant mechanisms. And this analysis can be done in the blood from the ctDNA, but also from the DNA of the tumor cells. And of course, we know that there are many of those aberrations in lung cancer, but also in colorectal cancer. We just heard that in the previous talk. So that has become more and more introduced in clinical studies. But however, not all the therapy resistance is actually engraved in a genomic aberration. There are also transcriptional changes that actually contribute to therapy resistance and therapy response. And these transcriptional changes require the analysis of RNA or proteins. They cannot be done on the DNA, of course. And here the CTCs make a nice offer. Let me show you just one example. In prostate cancer, of course, androgen therapy is the key. And researchers in Johns Hopkins have found out several years ago around Emmanuel Antonarakis that if you have a certain variant expression of the androgen receptor V7 expression, that these cells are more resistant to abiraterone or enzalutamide treatment. So we have here looked at different CTCs. Each line of the heat map is one CTC. We looked at different CTCs in a single patient. So we looked at intra-patient heterogeneity. And this heat map shows you there is quite a bit of intra-patient heterogeneity. Not two CTCs are really the same. But if you look, for example, for the ARV7 expression, which is here now highlighted, you see that 3 cells were positive, 4 cells were negative. And you can imagine that this heterogeneity can contribute to therapy resistance. We are also now able to combine this information of the ARV7 expression with the information of the phenotype of the CTCs. Rick Thompson has shown very nicely that the CTCs can be in different states, being epithelial, being hybrid, or being mesenchymal. And here in this small pilot study, we analyzed 17 patients with castration-resistant prostate cancer, and we could find 3 distinct CTC types. We could find CTCs which are epithelial, expressing the cytokeratin, CK positive, that were also positive for the resistant variant or negative for the resistant variant. But we could also find unusual CTCs that were not expressing the epithelial marker CK. These are probably the more mesenchymal CTCs that were positive for the resistant markers. So that also shows you to really integrate the information on the resistant marker with the phenotype of the CTC would be also quite interesting for the result interpretation. So another, of course, new target in prostate cancer is the PSMA, the prostate-specific membrane antigen. It is a diagnostic target in the PET-CT, but it has become also a therapeutic target in several clinical studies now. So it was interesting to see what is the expression of PSMA on CTCs. And we actually did that in the 4th channel of the cell search system. So you see that we have here cytokeratin positive cells, which do not express the leukocyte marker CD45. And in the last row, you see the expression of the PSMA. And you see in this patient, you have a striking heterogeneity. You have PSMA strongly positive cells, and you have also PSMA weakly positive and negative cells. And this heterogeneity may also really have an influence whether you are able to detect with your PET-CT the prostate tumor relapse or not, and whether your therapy will actually kill all the cells, because if they do not express the target, they may survive. Of course, we can also look at ctDNA to get information on druggable genomic aberration and resistance-inducing aberrations in prostate cancer. Just to show you one example, a study that we did together with Alan Heitzer and Michael Speicher in Graz. We looked at the plasma-free DNA in patients with castration-resistant prostate cancer. And the first line is a healthy control. You see, we don't pick up any changes in the healthy control, but the prostate cancer patients show several molecular changes in different genomic areas. And also they showed mutations in BRCA1, BRCA2 genes. And I think this information might be important for the use of PARP inhibitor therapy in prostate cancer. So the future is certainly to combine both worlds in a single tube to actually collect the plasma fraction, do the cell-free DNA extraction, and then look for the molecular analysis of the tumor DNA. And on the other hand, collect the molecular cell fraction, do an enrichment and capture of the CTC, pick the single CTCs, and look for changes in RNA and protein content, and then combine this information. We were also able recently to establish experimental models from the circulating tumor cells. And these experimental models and cell lines are probably interesting to understand the biology of CTCs, but also for drug screening to see which drugs really can kill circulating tumor cells. And this paper was just published recently in EMBO Molecular Medicine. And I like to show this slide to acknowledge all the collaborators that contributed to this paper. It's a real international paper. And just to show you a couple of slides from the paper, we could actually show by genomic CNA analysis that there were common aberrations of the cell line cells and the primary CTC. So the cell line cells were reflecting the primary CTCs. They were not just an artificial cell line. And we could also show that the copy number aberration profiles of the cell line remained stable in culture, which is also important. We then could actually inject the cells in immunodeficient mice, and we could show that the cell line can also grow at the primary site in the mice, and the cell line can also form metastasis in organs relevant to breast cancer, such as the bone, the liver, the lung, and the brain. And it was highlighted on the cover of the journal last year. We also did some drug sensitivity testing. For example, this was an estrogen receptor positive cell line from an ER positive patient that became resistant to hormone therapy at the time when the patient was actually admitted. Palbociclib or CDK4/6 inhibitors were not approved. So we were curious to see whether this kind of drug would work. And as you can see here, we treated our CTC cell line at the upper panel. And as a comparator, we treated ER-positive MCF7 breast cancer cells. And as you can see from the curves, both cell lines were actually sensitive to palbociclib and indicating that this is would have been a certain treatment for the patient. And this cell line is just, for example, available for international collaboration. So if you're interested, please let me know. So in conclusion, I think that CTCs, in addition to ctDNA and other biomarkers, provide complementary information for liquid biopsy. The assays need to be validated in an independent expert group. I think it's very important. There are many assays published, but I think that our network is always happy to test independently assays. And I think the key are now interventional clinical studies to demonstrate the clinical utility of liquid biopsy. We have to show that changing the therapy or the management of patient based on our blood test is really for the benefit of the patient. That's really what clinical utility is all about. Let me show you two studies that have shown clinical utility in breast cancer. The first one is from the French group. It is actually the SPIC randomized clinical trial. Here, the colleagues actually analyzed CTCs in hormone receptor positive, HER2 negative metastatic breast cancer. And they actually made the choice whether the patient got chemotherapy or hormone therapy based on the CTC count, and in the other arm based on the clinical decision guidelines. And here you see the Kaplan-Meier survival curve. You had on the right side, this is the most important curve, a group of patients where the clinical guidelines said that there is a low risk to progress. So they would have actually received only the hormone therapy, but the CTC count indicated that these patients were at higher risk. So then this group received the chemotherapy. And as you can see here, that the group in orange that received the chemotherapy based on the CTC high count actually had a better survival than the group that got the less aggressive therapy. So suggesting that the CTC count should be included in the decision algorithm for these patients. The second trial is a German multi-center trial, the called DETECT trial. Here we found many years ago that there was a discordance between the HER2 status of the primary tumor and the HER2 status of the CTCs at the time of metastatic relapse. in around 10 to 20% patients. So I think there was some tumor evolution over time between the primary disease detection and the detection of metastasis that has changed the subtype probably of these breast cancer patients. So we asked the question, if we treat these patients that are HER2 negative on the primary but HER2 positive on the CTCs with an anti-HER2 therapy, lapatinib, is that really for the benefit of the patients? And this result was just presented at the last San Antonio Breast Cancer Conference. And it shows you here that the patients that received the standard therapy plus anti-HER2 therapy with lapatinib had a better survival than the patients that received only the standard therapy. And this is now, I think, in preparation for publication. Another, I think, study that shows clinical utility of CTCs in breast cancer. So in order to actually get from the discovery to the clinical implementation, it's very important. I mean, these meetings, like your meetings, are important to spread the good word around. But we have also established in Europe, based on our Cancer ID network, a new network, which is called the European Liquid Biopsy Society. We have now 55 institutions from academia and industry. I'm coordinating that. And we are also reaching out across Europe, of course, to other continents. And if you are interested in that, our activities, please send me a mail. So the goal is to foster the introduction of liquid biopsy into clinical practice, encourage the interaction between academia and industry, provide a partner for regulatory agencies and patient advocacy groups, support the implementation of liquid biopsy tests into clinical trials, develop guidelines and provide trainings, and disseminate the knowledge about liquid biopsy. And I think these are very important goals, and we hope that that can help really to bring liquid biopsy into clinical practice. So at the end, I would like to thank the people at our institution here, the University Hospital in Hamburg, Germany. And you see, it's really a translational research project. We have research institutes on the left, we have clinics treating cancer patients on the right, and this interaction between both institutions is very important to get this translational research field going. These are the people in my institute. We are around 45 people from 14 different countries, so we usually speak English in our institute, and we are very happy actually also for new applications, and hope that the COVID period will be over one day and we can really travel and we can visit and we can host new people also in our institutions. I'm also very, very happy for the support, for the funding. We receive substantial funding from the European Union, the European Research Council, but also the other European Union programs, and also from the German institutions, the German Research Gemeinschaft, DFG, the Minister of Research, BMBF, and the German Cancer Ad Fund, the Deutsche Krebshilfe, which also has sponsored a postgraduate center at our Comprehensive Cancer Center. Thank you very much.
Thank you very much, Klaus. Indeed, it was really our pleasure, and the way you have touched base to the many of your projects. So it has given us actually a tremendous information that the kind of application we can use using CTCs and ctDNA. I could see many questions in the chat box. And Mani, would you like to take the question?
Yes, you go ahead, Prasad, it's fine.
So I have a couple of questions, Klaus.
Sure.
So CDX model, you know that we are battling with the success of this model. And I think my lab, we tried multiple times and we didn't succeed. So what is your success rate?
I think the success rate depends fully on the number of tumor cells. The success rate currently is really low. And you have to look for samples where you find at least, I would say, 50 to 100 CTCs in your blood sample, right? So I think if you start, let's say, on an unselected group of patients, the success rate is below 5%. But if you really go for patients with a very high CTC number, then the success rate is higher. However, these models are not really at present suitable for individual therapy decisions, right? I mean, the success rate is low, so the chance that you get a cell line from an individual patient to decide what kind of treatment is needed for this purpose, it's still too low. So these are just models right now, I would say, to understand the biology and also understand the drug response of CTCs, you know?
So
we are working really on increasing the efficacy of setting up cell lines, also temporary cell lines, let's say. And we are working on that by increasing the blood volume and increasing the capture rate. But this is still work in progress.
Do you think the clonal population or the clonality of the cell population matters?
Oh yes, I think that what we have seen is that the CTCs are polyclonal. They are derived. I mean, they have different genomic aberrations, you know. We are just in the process of trying to identify which are the bad guys, so to say, you know. Yes. And the bad guys that initiate metastasis, it could be a certain genomic aberration pattern, but it could be also a certain expression pattern. So we have done a full-scale RNA sequencing of these cells, you know, and we are now comparing also those cells that are able to grow in the mice compared to those cells that we injected in the mice to see really what is the tumorigenic subclone of these CTCs. Yeah. Mm-hmm.
I just have Prashant, I just want to ask one question. So along the line, what you just said. So in your presentation class, you just said that you saw the primary tumor being HER2 negative, but you saw CTC which are HER2 positive. So how would you explain the biology of that? How do you see HER2 positive CTCs? I don't know how prevalent they are. They are just few or more in the CTC. If you do, how to explain that?
Well, there are two explanations. The one is it's a pre-existing clone, you know, a very small clone that was maybe overlooked by the primary tumor analysis. And the second explanation is that these cells just appear under the pressure of the adjuvant therapy and the hormonal therapy that goes for many years, you know. And we are just currently trying to figure out what it is, you know. So we are also, it's also very interesting that not all of these HER2 positive might have an amplification. There are also very nice data by, you know, Dan Haber's group published several years ago that it could be also transcriptional switch. So we are just, I would say, working on the mechanisms, yeah. Mm-hmm.
Thank you.
So in your mice model, how close correlation you found between the primary tumor and the metastatic regions? By any chance, if you correlated any transcriptomics data or anything with the genomics, and how closely the correlation was?
Well, we are just in the middle of analyzing the metastatic, let's say, metastasis in the mouse and compare them with the primary tumor. I mean, so I cannot tell you right now. But I know that the comparison of the primary tumor with the metastasis has been done for many, many years with humans, you know, in patients. And it's quite depressing that there were not so many differences, you know. I know that Laura Van der Weer and others have done that. They compared primary and metastasis in breast cancer. And what they actually found is that they could identify which tumors were from the same patient, so to say, you know. So we will see. We will continue to do that, and but it's too early to say right now. We're also developing drug-sensitive sublines. We're deriving different sublines going to different organs to really see whether there are organ-specific changes. So this is all just work in progress. It will keep us busy a few years. Yeah. Mm-hmm.
Excellent. So there's a query from Dr. Hima Bindu. Could you please comment on the feasibility and utilization of CTCs to develop organoid model to facilitate drug screening?
Yeah, I think this is really something we are also doing. I think that, and other people too all over the world, you know, using organoid models also to increase the efficiency of cultivating the CTCs and then doing the drug screening. That is certainly a short-term goal that we pursue. Absolutely. It's, I mean, it will also, we have to show, of course, to what extent then this drug screening mirrors what is going on in the cancer patient, but it's certainly one of our projects, yeah. Mm-hmm.
A couple of queries from Dr. Shyamla Maheshwaran. Excellent talk, her comment, and there are two queries from her. Does the ratio of CTCs with ARV7 and wild type 80 have any clinical value?
So when you
well, we have. I mean, these data have been actually published largely, I would say, by Emanuel Antonorakis group, for example, in the JCO 2017, and there are follow-up papers also by Howard Scher's group, you know, doing it at the immunocytochemical level. And I think that people have really shown that if you have CTCs that express the V7 variant, that these patients have a bad response to abiraterone and enzalutamide as compared to the patients that have CTCs with the wild type. And I think the ratio, the exact ratio, I would say, has not been determined. These were just data where the data were actually dichotomized in V positive or V negative, you know. And whenever there was a V positive signal, it was called V positive. But I think we will probably take now also the intratumor heterogeneity into account in future studies. Yeah. Mm-hmm.
Yeah. Dr. Shyamla, please feel free to ask your other question. Please.
So the other question that I had was, I really liked your CTC cell line model and the experiments that you had done with it. The question I had was whether these are ER positive.
Yes, the cell line is ER positive. It's also stable ER positive for a few years now, which is really nice. We also in the paper have analyzed a bit more whether the ER signaling axis is fully intact. It is not, okay. So there are some perturbations in it, but the cell line, I mean, the protein is fully expressed and it's expressed over years. So it's one of the few ER positive cell lines, yeah. Mm-hmm.
Yeah. Sevanthi, would you like to ask the question, please?
I think we should let Dr. Kasi, he is posted.
I was, going to comment, Dr. Patel, that in terms of the HER2 discordance and heterogeneity that you brought up, we're seeing increasingly in our upper GI cancers too, that post anti-HER2 blockade, the places where they rebiopsied the tissue, in at least 30% of the cases, they had lost HER2 expression. So I think the future probably is going to be factoring tissue and liquid both. I think the temporal as well as the intratumoral heterogeneity, HER2 is a great example beyond breast in GI cancers too, where probably you factor CTCs or multiple tissue biopsies. There's discordance we see all the time.
Yeah, no, I think this is a very good comment, and I made some notes during your talk on. I mean, I was really excited to see it's really 30%. You know, I mean that it can emerge in such a high incidence. So there might be also something mechanistically in it. You know, that a HER2 positive tumor cell may have a real significant advantage in different tumor types. You know, yeah. Mm-hmm. So, very interesting.
I just have a quick question. Sorry, just hopefully the last one. So again, Dr. Kashi and you, both of you showed a nice data showing that the ctDNA is able to separate the patients into two groups, and particularly with respect to clinical outcome, that's spectacular. So now somebody also asked a question in the chat, like, can you add additional biomarker on top of that so that you can even separate it even better? Number one. Number two, can you also add additional marker on top of the ctDNA itself? Like, right now you are using a set of biomarkers to separate them into two groups. And can you add more biomarkers on top of that? And Seb, would it help?
Yeah, no, that's a very good. It's a very good remark and question. That's exactly what we are doing now in a new trial. You know, that we are setting up and. We are now in the final round of getting governmental support for that. And there we really combine CTC, ctDNA, and protein markers, and maybe additional ones to further separate the curves. Because in the papers we have to keep in mind the ctDNA paper are not really thousands of patients. So we do a study now with more than 3,000 breast cancer patients. Okay, because we really need high numbers to be convinced, right? And the second thing is, yes, the ones that keep ctDNA positive have a bad prognosis, but usually also there are some which are negative and they also go a bit down, right? I mean, and of course, we don't want that. We would like to have a better separation, let's say. And we would also, of course, see that the number of ctDNA positives also in the colon trial, if you look at the number of patients in the ctDNA positive arm, number is very low, really, So, I mean, the risk is actually higher, I would say, So yes, you make a very good point. These studies are very, very encouraging, right? But I think we need follow-up studies on larger numbers of patients, larger cohorts, and include maybe more markers. I definitely agree with you here. Mm-hmm.
Thank you.
Mm-hmm.
Yeah, Sevanthi, would you like to take your question?
So I have a question from Dr. Mandar Kulkarni. He says, if HER2 is a less faithful marker for choosing therapy based on CTCs, are there other potential more faithful markers emerging from NGS work?
No, I think the HER2 is a faithful marker for the CTCs. That's what our study did actually show, because when we treated the HER2 positive CTC patients with HER2 positive CTCs with anti-HER2 therapy, they had a profit from it. So no, I think it's a faithful marker. But of course, I mean, we need also additional markers because, I mean, not every cell is, of course, HER2 positive. And we also need to look, for example, we have looked at the estrogen receptor expression also in the same way, and we also saw discordance in some patients of the ER expression. And I'm sure everybody knows that there is emergence of ESR mutations, over time. So I think we need to do the restaging, whether we do it at the ctDNA, CTC, or maybe biopsy of metastasis. But we need to do a restaging, because if there are 5 years between initial, let's say, primary tumor resection and the occurrence of metastasis, or sometimes even more than 5 years, the tumor type might have changed, right? And we just categorize the tumors at the beginning of a therapy when we do the biopsy or the surgery, but we have to do a recategorization at the time of relapse. That's what we need to learn, and that's really new. Mm-hmm.
That's great. That's great. I will ask a purely clinician's question here, Klaus. So as a medical oncologist, this is a dilemma I face all the time. As I treat my patient and I see that the ctDNA is showing lack of response to therapy, mm-hmm. I know this treatment is not working. Mm-hmm. But then there are so many more, and the patient's condition may be declining, and I have to choose the right one. Would you think we are ready to utilize CTCs as a therapy strategy planner? Or is it something that is being looked at for planning a strategy for the next line of therapy? In terms of choosing?
I think we really have to do the right clinical studies. I think, we have to do this clinical utility study. We have to do interventional studies, right? It's very important. We can, of course, right now, we may use it in individual patients because we don't have this data and the patient is there, so we have to do something, okay? But I would say we have to initiate these interventional trials to really see if A is not working and I use B as a therapy, based on the ctDNA or CTCs. Is that really working? And that's what we did with this, DETECT trial, and that's what the French did with their STIC trial. And we need this information. Another way, probably in the intermediate time, what is also a good idea is if we do 2 measurements of CTC or ctDNA within, let's say, 4 weeks, 3 to 4 weeks, And if we see that it's going up or down or not changing, this kind of monitoring information within, let's say, a few weeks, that can also give an indication whether the next line of therapy is going in the wrong direction or not, okay? And that's something we have seen several times. If there is a steep decrease in the CTCs within 4 weeks, we feel a bit more relaxed that our next therapy is doing better than the previous one, right? So this might be also this monitoring, let's say, with 2 blood samples within a few weeks, may give you an idea whether your next therapy is working or not.
And you do them longitudinally?
Yes, yes, and that's a nice thing with the blood. You can take blood, you know, every few weeks or every week. I mean, that's a good thing about it. Yeah.
Excellent.
One quick clinical comment I was going to make is, interestingly, just November of 2020, one of the tumor-informed assays, the Signatera from Natera, got Medicare approval for immune therapy response monitoring. So that was based on the Nature Medicine paper where they had looked at the ctDNA clearance or the delta positive versus negative in patients who were getting Durva plus Tremi, the AstraZeneca's immunotherapy drugs. And based on that, Medicare actually in the US has given draft coverage for using that assay to, you know, the ctDNA kinetics between baseline and 6 weeks apart as a way to maybe gauge immunotherapy response. So it's already getting into the clinic as we speak.
Yeah,
you know.
That's very good. And I know that Howard Share, they have really filed also approval for CTC 0, they call it. That means if it drops to 0 CTC during the treatment, you know, in prostate cancer, that's a quite good sign that the treatment is working, you know. So this is also a kind of surrogate or early response marker. And I feel this is good. I'm happy to hear that. We need this information, these tools, yeah.
So there may be the last question from Barnali Dev. She's a PhD student, and I could see the synergy of her thought, and I'm also now interested to understand what she's asking. What was the phenotypic expression of the CTC cell line? Did they exhibit epithelial, mesenchymal, or the hybrid phenotype?
That's a very good question, and I really have to refer you fully to the paper. We had epithelial markers like cytokeratins that were EpCAM positive, right? So from the first Western blots, so to say, you know, and also from our RNA sequencing, it was really called, or it is called, an epithelial cell line, okay? We also did a RNA sequencing. We looked at Jean-Paul Thierry's score, you know, and he is also a co-author. So that looks very epithelial. But then we looked at, let's say, the cell shape, and we looked a little bit at the cell structure, and they also grow in an adherent and non-adherent fashion. And so we saw also some, let's say, signs of mesenchymal features in it, you know. So we had a long discussion. Is it epithelial? Is it hybrid? It's probably epithelial, but it has some tendency to be hybrid. I would call it this way, yeah.
Yeah. So we do have done the EMT scoring on the CTC cell, CTCs actually,
yeah,
and compared them with the primary tumor. And what you are saying, the similar kind of observation that most of them, the scoring lies between the EMT, so hybrid phenotype, and some of them are definitely mesenchymal and epithelial. So yeah, I think more data needed until we conclude something.
Yeah, yeah.
Thank
but
you
I should say our cell line is EpCAM positive. We should say it.
So thank you very much, Dr. Pantel.
It is my pleasure, really.
For enlightening us, for teaching us, and we really look forward to have many more collaboration, association, and a meeting point so that we could learn more. And I appreciate your time. I really appreciate your time. Thank you very much.
Thank you very much for inviting me to this excellent meeting, really. And thank you also for all the fantastic questions. Thank you.
Okay, so now we'll move to our next speaker. And this is none other than Professor Shyamala Maheshwaran. And I would again say that it's my really, really great pleasure to introduce her. And I think, again, the people who are working in the area of liquid biopsy, I think they may not need any introduction about Shyamla's work. But yeah, so she's currently a professor of surgery at Harvard Medical School. She's currently in this, also the scientific director of the Center for Cancer Risk Assessment at MGH. She's co-director of the CT Circulating Tumor Cell Laboratory at MGH and the co-founder of a company, TelBio, that will commercialize CTC iChip technology. She has published numerous publications, very, very high impact factor journal, and expertise and training in most aspects of tumor biology to successfully direct the proposed project. Her lab actually characterized the contribution of oncogenic cues, tumor microenvironment derived signals, EMT, tumor heterogeneity to breast cancer progression and therapeutic response. I won't take much time, and this is all to you, Dr. Maheshran. And again, thank you very much for accepting our invitation. Thanks a lot.
Thank you, Prashant. And I'd like to thank all of you for this invitation. And I also have to say that I'm really proud to be a part of this great symposium that has been put together by Prashant and Swanti and Mani. And we have heard some really great talks over the past 2 days, all highlighting the importance of liquid biopsy in revolutionizing the care of cancer patients. So basically, today what I want to do is to show you some data that illustrates the contribution of epithelial to mesenchymal transition to different levels of tumor heterogeneity. So tumor heterogeneity, and we have heard about this, and I hope that the data that I'm going to be showing you today will actually sort of help us provide some insight into the molecular evolution and drug responses that we have been hearing about in all these talks given over the past 2 days. So tumor heterogeneity comes in many different flavors, and I've just highlighted a few here. So you can have differences in cell lineages where epithelial tumor cells coexist with mesenchymal tumor cells. It can also arise from differences in proliferation where there are proliferating tumor cells coexisting with quiescent tumor cells. And we can also have differences in growth factor signaling. In fact, using circulating tumor cell cultures established from metastatic hormone receptor positive breast cancer patients who had received multiple courses of therapies, we showed that these tumor cells have a HER2 positive population and a HER2 negative population, and they're highly plastic. They can convert from one state to another. They have different tumorigenic properties and drug sensitivity profiles. And in addition, we also have this genomic heterogeneity that we have heard throughout the talks in the past 2 days, where you have various populations with different mutations rising and falling depending on therapy. And these are not actually isolated states. In fact, there's quite a bit of interactions between them. You find that these HER2 populations are in fact highly proliferative, whereas the HER2 negative population was found to be quiescent. And that actually sort of influences their tumorigenic properties and drug responses. So today I want to share 2 stories with you. One of them is basically a summary, which shows that epithelial to mesenchymal transition is incompatible with simultaneous proliferation. And when these 2 processes occur simultaneously, there's extensive mitotic defects resulting in genomic diversity. So, and in the second talk, which I will go into in quite a bit of detail, I'll show you data that illustrates using single cell proteomics that epithelial and mesenchymal populations have differential kinase activities, and that this influences their kinase drug sensitivities, which might shed some insight into why EMT contributes to drug resistance. So we had shown a couple of years ago using RNA in situ hybridizations using probes to mark epithelial and mesenchymal states. We applied this technique on circulating tumor cells that were isolated from metastatic breast cancer patients. And it showed us that EMT was not representative of two distinct states, but instead is a continuous process with multiple intermediate states present in these CTCs. And when we actually looked at these CTCs carefully, we found that these mesenchymal CTCs often presented as either binucleated cells or cells with micronuclei, and this fraction was much higher in the mesenchymal CTC population compared to the epithelial population, suggesting that these mesenchymal cells might be experiencing some degree of mitotic stress. So to understand this, we actually turned to an in vitro system where we treated epithelial tumor cells with TGF-beta, TGF-beta being a very potent inducer of EMT. And we found that, in fact, induction of EMT was associated with the appearance of these binucleated cells and cells with micronuclei. And we actually showed that this was because these cells were experiencing extensive mitotic defects, which led them to have extensive genomic instability. When we karyotyped these cells, we found that they were often polyploid compared to the untreated cells, which were very close to a diploid karyotype. And as a result, they are very tumorigenic. And in fact, when you do single cell karyotyping of these tumors, you can see that these tumors are highly heterogeneous based on their genotype compared to the initial cells that we started with, which were which exhibited a very tight diploid karyotype, suggesting that EMT was basically not only a plastic process, but was contributing to genomic heterogeneity. And when we actually induced EMT in non-proliferating cells, basically none of this happened. So suggesting that EMT and proliferation were incompatible with one another. And this is to be expected because both EMT, which is associated with increased migration and mitosis, basically involve extensive changes in the cytoskeleton. And as a result, when they occur simultaneously, it is incompatible, or it's they actually sort of send competing signals to the cell, and the cell gets confused and actually results in having these mitotic defects. And that eventually contributes to the genomic heterogeneity and the molecular evolution that we actually see in tumors over time. So in the next segment, we have actually heard about genomic tumor heterogeneity throughout many of the talks that we have heard. But most of these studies have been done based on single cell RNA sequencing or sequencing with NGS to look at the evolution of the mutational profile. However, proteomics, single cell proteomics to understand signaling pathways that actually evolve over time as the primary tumor metastasizes to the various distal sites like lung, liver, and bone is not fully understood, partly because of the limited sensitivity of the single cell proteomic approaches that are currently available. So what we did was we actually applied single cell proteomics, mass cytometry, to look at the signaling heterogeneity that exists in these isogenic signaling compartments in a mouse tumor metastasis model. And we used two different mouse tumor metastasis models, both for prostate cancer. One was derived using PC3 cell line. This is a human prostate cancer cell line, P10 deleted, and it is AR negative. CE114 is also a prostate cancer cell line, but it was derived from the mouse, which had a P10 deletion, but it retained all the androgen responsiveness. So we, first of all, we actually sort of came up with a 29 antibody panel to interrogate kinase signaling, the signaling pathways in single cells. And this included the various intermediates that queried the PI3 kinase ImmTOR pathway. They are all color-coded here. MAP kinase pathway, the RTKs, all of which are shown here, the cytokine signaling pathway, and of course, lineage, we had EpCAM and cytokeratin as well. So what we did was we first of all wanted to validate these 29 antibody panels. So we used an in vitro system where we treated these PC3 cells in vitro with a dual inhibitor that blocks the PI3 kinase and ImmTOR pathway, BEC235. BEC235, and treatment of PC3 cells in vitro with BEC235 basically blocks their proliferation. And when we did single cell mass cytometry in over 650,000 individual PC3 cells, we found that treatment with this BEC235 suppressed the expression of phospho-S6, 4EBP1 and mTOR intermediate. It also suppressed phospho-AKT473, but not 308. And there was no effect on the expression of GSK3 beta. So we actually validated this using a Western blot of untreated and BEC235 treated PC3 cells. And you can see that these findings basically mirror the data that we actually obtained with the single cell mass cytometry. There is a suppression of phospho-S6, 4EBP1, AKT473 is suppressed, but not 308, and you don't really see any change in GSK3 beta. So we then applied, after having validated this in vitro, we applied this to this mouse tumor model. The PC3 cells were inoculated into the mouse prostate. They grow and they spontaneously form tumors in the lung, liver, and bone. So we collected all these primary and metastatic tumors, treated with collagenase, made single cells, and did mass cytometry with that 29 antibody panel that I actually just described. And we also collected CTCs from the blood of these animals. So what did we see? When we did a Visney plot of all the data that we had generated, we found that basically the primary tumor was highly heterogeneous, creating 2 fractured islands. And interestingly enough, the CTCs basically overlapped with whatever we found with the primary tumor. And here you find that the primary tumor cells also overlap to some extent with the metastatic tumor cells that come from the lung and the liver. Now, interestingly, the bone basically fractured away from all these primary and metastatic tumors and formed its own island. And when we actually looked at the signaling pathways in the bone and compared it to the tumor cells that were isolated from the various compartments, the primary tumor, the CTCs, the metastatic tumors from the lung and the liver, and here's the bone metastasis, we found that there was a selective upregulation of RTK signaling defined by EGFR and CMET, a selective upregulation of mTOR-PI3 kinase signaling, phospho-S6, 4EBP1, AKT, and phospho-GSK3 beta, and there was also an increase in the MAP kinase signaling, all limited to the bone metastasis compared to the tumor cells that were coming from all the other compartments. So the CMET and the EGFR are very interesting because there has been multiple publication where folks have actually looked at the bone metastasis in castration-resistant prostate cancer patients and compared it with the primary tumor. And over 80% of bone metastasis in prostate cancer patients have increased expression of the c-Met protein. And so these single cell mass cytometry in this mouse model independently validates the findings that have been already published. And the same is true for EGFR. However, you find that, c-Met is not actually, or these RTKs are not upregulated compared to the primary tumor in the tumor cells that are residing in the other tumor compartments. Despite being isogenic, they seem to be about the same. At least they're not significantly different compared to what you see in the primary tumor in the other metastatic tumor compartments. Now, why is this important? Actually, based on the finding that over 80% of castration-resistant prostate cancer patients were expressing high levels of c-Met in their bone metastasis, there was a trial that was initiated with cabozantinib, which is a c-Met and a VEGF receptor inhibitor. And interestingly enough, this inhibitor wiped out the bone metastasis and improved progression-free survival without any effect on overall survival, suggesting that it may have actually eliminated the metastasis in the bone, but might not have actually had any too much effect on the tumor cells residing in the other compartments. So our data is showing that it's very important to take into account the tumor microenvironment, which is actually also influencing the protein signaling and contributing to changes in signaling events that happen at these different sites. So we then assigned a PI3 kinase ImmTOR index, where we summed up the intensities of the various intermediates, and shown here as is a different illustration compared to the bar graph I showed you. There's an increased PI3 kinase ImmTOR index in these in the bone metastasis in the PC3 mouse model that was also confirmed with the CE1-4 mouse prostate cancer model, again, which had an increased PI3 kinase ImmTOR index in the bone compared with the primary tumor. We validated this using immunohistochemistry. As you can see here, compared to the primary tumor, there's increased staining of phospho-S6 and 4-ABP1 in the bone metastasis. The quantification of it is shown here. And we then wanted to see whether we could actually extrapolate this to prostate cancer patients. Now, prostate cancer predominantly metastasizes to the bone, but it is very difficult to actually get biopsies of the bone. However, these patients actually have CTCs in their blood, as we have heard in the various talks. And these CTCs can serve as a proxy for the bone metastasis that is present in these prostate cancer patients. So when we compare, and we had actually isolated 77 single CTCs from 13 castration-resistant metastatic prostate cancer patients. And when we looked at the single-cell RNA sequencing for ImmTOR and PI3 kinase signatures, we found that they were significantly elevated in the CTCs compared to the primary tumors. So we then wanted to see how these signaling changes would affect drug responses. So basically, this was the PC3 model. We treated the mice with BEC235, that is that dual inhibitor that blocks PI3 kinase and ImmTOR pathways. We collected the tumor cells from these various tissue compartments, and of course did mass cytometry. And to compare, we actually also used GDC0941, which is an inhibitor of the PI3 kinase. And you find that basically GDC, while the dual inhibitor blocks primary tumor growth, GDC0941, which blocks just the PI3 kinase pathway, has no effect at all on primary tumor growth. So now looking at the single cells that were derived from the untreated and the treated primary tumors and CTCs, looking at after doing a single cell proteomic analysis on the tumor cells that were obtained from these mice, we actually looked at the PI3 kinase ImmTOR index, and there was a suppression of the signaling pathway consistent with the growth inhibition that was seen in the mouse. Now, interestingly, GDC0941, which had no effect on tumor growth, had absolutely no effect on the signaling pathways in the tumor cells that were interrogated from these two different tumor compartments. Now, what about the metastasis? So I had told you that the bone metastasis had a much higher level of PI3 kinase ImmTOR signaling compared to the PI3 kinase ImmTOR index in any of the other tumor compartments. And consistent with that, we found that the bone metastasis was exquisitely sensitive to BEC235, with none of the animals having tumors, at least having any tumors after treatment, compared to 75% of the control animals showing tumors in the bone. And when we actually sort of looked at the bone, we couldn't find any cells that could actually be used for further analysis. However, interestingly, the liver, we were able to still see some lesions. We could get adequate numbers of cells from these lung and liver metastases. And when we interrogated these cells, there was absolutely no change in PI3 kinase ImmTOR signaling pathways in these metastatic lesions, suggesting that, basically, while this dual inhibitor was kind of wiping out the tumor cells in the bone, it had absolutely no effect on the tumor cells that were residing in the lung and the liver, suggesting that the tumor microenvironment must play a role in regulating these drug responses and the pathways, despite all of these tumors being isogenic and coming from a PTEN deleted original primary tumor. So we then went on to actually, because we had a lineage marker EpCAM, we stained our tumors with EpCAM and saw in the untreated animal, the tumor cells were predominantly epithelial, and there were a few clusters of EpCAM negative mesenchymal cells. And upon treatment, when you harvested the cells and the tumors and stained them with EpCAM, basically the tumor cells were EpCAM negative, indicating that they were mesenchymal, and that is also shown here. The EpCAM IHC index shows a decline in EpCAM activity, and the mass cytometry confirms that, where the percentage of EpCAM low cells increase in the CTC compartment and in the primary tumor. But what was interesting was when we went back and looked at the data, here's the data from the primary tumor and the CTCs. So here are the EpCAM low population and the EpCAM high population. And what blew us away was that this EpCAM high population had very high levels of kinase activity. It had a high RTK signaling, and also it also had a high PI3 kinase IMTOR activity compared to these EpCAM low mesenchymal cells. And upon treatment with this drug, basically you find that there is a decline in the number of epithelial cells as expected, and an accumulation of these mesenchymal cells with this low kinase activity. And this was not only true for the primary tumor compartment, we also saw this in the CTCs, where there was a coexistence of a RTK high, PI3 kinase IMTOR high epithelial population coexisting with a kinase low mesenchymal population, and the entire thing switching over to a mesenchymal state with low kinase activity upon treatment with this dual kinase inhibitor. So we then wanted to extrapolate this to our CTCs. We had actually single CTC RNA sequencing that we had derived from prostate cancer patients, as well as 2 sets of breast cancer patients. So we applied an EMT signature on these CTCs, and we saw that there was indeed a population that was that was mesenchymal and epithelial. And when we actually looked at the PI3 kinase IMTOR signaling in these CTCs, you find that the PI3 kinase IMTOR signaling is much higher in the epithelial CTCs compared to the mesenchymal CTCs. And I've actually illustrated that a little bit better here. These are the single CTCs from the metastatic prostate cancer patients, and you can see that there is when you apply an EMT signature, you can identify the mesenchymal and the epithelial populations. And when we look at the PI3 kinase IMTOR signaling in these CTCs, it's much significantly higher in the epithelial population compared to the mesenchymal cells. And again, we had 2 metastatic breast cancer data sets from different patients. These CTCs, we could identify epithelial and mesenchymal CTCs, and we found in both data sets, the PI3 kinase IMTOR signaling was much higher in the epithelial population compared to the mesenchymal population. And we had survival and progression data on data set 1, and we found that patients who had a higher level of PI3 kinase signaling often had a poor overall survival and lower time to progression from the previous therapy. So basically, to summarize, I think while there have been tremendous amount of effort on looking at RNA sequencing, single cell RNA sequencing data to look at tumor heterogeneity, single cell proteomic analysis to look at the differences in signaling pathways in tumor deposits at various sites in a patient has been limited by the limited sensitivity of the currently available technologies. So we applied this mass cytometry method to a mouse tumor model where the primary tumor, a P10 deleted isogenic tumor cell, seeds at these different distal sites. And sort of continues to grow. And when we actually looked at it, we found that there was quite a bit of heterogeneity in kinase signaling. We found that the bone cells basically had the highest level of PI3 kinase IMTOR signaling. And there was also intratumoral, so creating a intertumor heterogeneity. There was also intratumoral heterogeneity, where the epithelial cells with the high kinase activity coexisted with a mesenchymal population that was low in kinase activity. And that this actually explains their responsiveness to drugs where, of course, these bone cells are effectively eliminated when you treat it with BZ235. Whereas when you look at this mixed population, of course, these mesenchymal cells are a lot less sensitive to the kinase inhibitor compared to the mesenchymal cells, which might explain the mechanism underlying EMT's ability to contribute to drug resistance. So basically, I have actually shown you two models where epithelial to mesenchymal transition thought to be a plastic process without any sustained genomic changes can actually, in fact, contribute to genomic heterogeneity when it occurs simultaneously with proliferation. And in addition, it also is able to contribute, depending on the tissue microenvironment, or even in intratumoral heterogeneity, where it actually has differential kinase activity, which is actually defining the ability of these cells to respond to drugs. With that, I like to acknowledge the people who did this work. This work was done by a postdoc. The single cell proteomic analysis was done by Murat Karabakh and Eric Zhang. And the genomic heterogeneity work that I showed you very briefly was done by Valentine Comey, and she was supported by many postdocs and other technicians. And also we have, I collaborate closely with my colleagues, Daniel Haver, Mehmet Toner, Ravi Kapoor, and many of these people, and David Ting. Most of the bioinformatic analysis is done by our bioinformatic team. And with that, I'd like to close and take any questions that you may have.
Thank you very much. I would say it's a mind-blowing data because anybody who's working in the area of proteomics, they could only dream to do. And I think you may be the first in the world. I don't know. I have never heard. Means we keep dreaming that we will do one day proteomics on CTCs. Knowing the fact that there's a limitation of the proteomics with the CTCs, and you cannot amplify the protein. And I think this is one of the bottleneck. Very quick thing which comes in my mind: why not you started on the patient sample instead of mice?
So here's the issue. I think with the patients, you can get sufficient. So when it's a highly limited technology. So you need large numbers of cells to be able to make a decision. And somehow in the mouse, you can get adequate numbers of CTCs to be able to do these kinds of studies. Whereas in the patients, oftentimes you get one cell, you might get 100 cells, depending on the patients. So to get a patient with 100 cells and to kind of cut your losses and analyze the data in a meaningful way is very difficult, which is why we use the mouse model as an investigational tool and then interrogated our data in the RNA sequencing panel that we had actually generated in the lab. Because I think that initially we would never have even thought of actually looking at that. So we were informed by the basic biology to go into the patient samples and address that. And lo and behold, it was a very striking difference of signaling in those two populations.
So we didn't observe much more overlap between the primary tumor and the CTCs, proteomics.
Yes.
Don't you expect other way around? Are we not expecting that there should be overlap?
So I think that, one of the things that you have to remember is I just used the 29 antibody panel, specifically interrogating what I wanted to look at. And I was lucky to be able to see something. I mean, we didn't know what we were going to expect.
And how did you pick these 29 antibodies? I just maybe missed it.
No, I think that, because there's so much interest in the RTKs, there's so much interest in the PI3 kinase mTOR signaling, especially with all these dual kinases coming up and the new trials that are going on with the androgen inhibitors and a combination with the PI3 kinase inhibitors. So we decided that we would actually choose these pathways. But on the other hand, that is not to say that they are identical. For this particular 29 antibody panel that interrogated a couple of different signaling pathways, the CTCs and the primary tumors overlapped.
Okay. I'll quickly ask my last question, and then I'll just start reading. There are multiple questions on the chat box. Epithelial high kinase, mesenchymal low kinase, both in CTCs and the primary tumor. So maybe this, so can I correlate that CTC having the epithelial characteristics are more aggressive? And that may be the reason when we talk about the metastasis or the CTC reaching to the distant organ, they may colonize because they are more aggressive. Do you think that could be the explanation?
No, I think it's a double-edged sword. I think that, the fact that the epithelial cells have a higher level of PI3 kinase mTOR signaling might actually sensitize them to a dual inhibitor that hits it. But on the other hand, if you actually look at the mesenchymal population, which has a lower level of this kinase activity, they are quiescent. If you look at the proliferative signature, which I didn't highlight, but it was on that heat map, they are much less proliferative, so that they become very resistant to any drug that hits them, right? So not only do they not respond to the kinases, but just simply because they are proliferating at a lower rate because of all these signaling pathways being much lower in them, it makes them resistant. So I think it's a new way of looking at them as to why they are, first of all, proliferating slowly and why they are resistant. It just gives you some insight into why.
Excellent. So I'll just take some of the questions. So Rick had asked, does base 235 cause EMT per se or selective kill EP cells?
Can you just repeat that question?
Rick, please address your question. Rick, can you please?
Yes, sorry. Thanks, Shamala. Really fantastic talk. Just the question was, do you get EMT when you use the BZ235? It seems you will selectively kill the epithelial cells from what you said, but does it also drive EMT?
So if you actually look at the growth curve, it appears that the tumors just actually stay. There's a very strong suppression of tumor growth, but eventually the tumor begins to grow. And we actually did a staining of the tumors as the tumors were just beginning to take off, and they are primarily mesenchymal. So I think that what happens is that BZ basically kills all the cells. But these mesenchymal cells, perhaps they change in some way and they begin to grow. Does BEC cause EMT? I don't have any evidence to say yes or if it doesn't. I don't think, based on sort of quickly evaluating all the data in my mind, but that's an interesting question.
Okay, thank you so much. Really nice.
Klaus, would you mind please asking your query?
Sure. Well, that's actually an excellent talk. Thanks a lot. I was just wondering, did you have the chance to look at lung, liver, bone metastasis and compare them in prostate cancer patients?
No, we haven't. I think it has been very difficult to actually get those samples. But I think that given some of the interest in treating these castration-resistant prostate cancer patients with combinations, right, with AR targeting therapy and some of the PI3 kinase I think that would be very, very important to do.
Yeah, maybe we can collaborate on that. We have a special group of clinicians and patients focusing really on, let's say, early progressing prostate cancer patients that also make metastasis in these, visceral organs. And I know that the colleagues sometimes take biopsies, of the liver at least. And we also have a rapid autopsy program. So maybe that could be something,
And I can actually send you the paper that we have just submitted. So
yeah,
if you are interested in taking a look at it.
Cool. No, very nice, really. Thank you.
Thank you.
I'll take the last question from Mohit Kumar Jolly from Indian Institute of Science, and he works on EMT. So what Mohit is asking, is the inhibition of cell cycle during EMT a progressive decreasing feature, or is there a sudden transition at some point? How does the proliferation of hybrid EM cells look like?
So I think that, these are basically in vitro models. And I think that, what happens is that, so one of the pieces of data that I didn't actually show is this. So when you actually sort of induce EMT in cells, what happens is that EMT is associated with increased migration. And the nucleus is the most rigid structure, which actually needs to soften a little bit to facilitate movement. And what happens is that is exactly what happens. These EMT-inducing agents soften the nucleus by decreasing the expression of the nuclear envelope proteins. However, the proliferation requires these nuclear envelope proteins because they moonlight in driving proper mitosis. So what happens when the two happen at the same time? Basically, the cells are, how do you say, are at an advantage to migrate, but they are not at an advantageous state to proliferate because the nuclear envelope proteins are down. So the moment it initiates, I believe, even when you get into the hybrid states, you have already started propagating these conflicting signals. I hope that I answered your question.
So I just have one quick question. If that's done, can I ask?
Please, Mani, please.
So Shyamala, wonderful talk. Just my quick question is, in one of your presentation, you said that you separated the EMT E versus M, and then that signature correlates with respect to EMT. But then you looked at a PI3 kinase mTOR signaling. For whatever reason, you see mTOR signaling is low in a cells undergone an EMT or a mesenchymal, which is kind of opposite of what one would expect, because the cells undergone an EMT should be more resistant to survival, so therefore you should have the survival pathway should be higher in these cells than the E line. So therefore, have you thought of separating them into E? We have been talking about the hybrid EMT cells being in a middle stage versus a mesenchymal stage. Have you thought of separating them into 3 groups rather than 2 groups, and then ask, would the mTOR signaling could be high in the middle group?
That's a very good point. So let me take the first question first. I agree with you that it would make sense because they survive, that they should have increased PI3 kinase mTOR signaling. But I also feel that they are less proliferative, right? So what happens is when they have these survival pathways down, they get into I won't say quiescent, but because we didn't query a quiescent signature, we just looked at the proliferative signature, but it allows them to escape drugs, right? Because so as a result, I think that the fact that they suppress this and suppress proliferation gives them an advantage to survive, if that answers your question. The second question that's a very good point, because I think we had many more CTCs than we could actually separate out E and M. So there was an intermediate population that we never really looked at because we wanted to keep it clean. For example, there was one set we had 135 CTCs, another one we had like 106 CTCs, but we could cleanly separate out only about 2/3 of those into E and M. We left out that middle section because it didn't even occur to us to look at, but that's a very good point. We should take a look at that.
Thanks.
I have a couple of questions actually.
Yeah, please.
And I'm going to utilize the opportunity, and what an honor, Dr. Maheshwaran, to have you here today, despite all the challenges, and thank you so much for gracing us. So I have a question about PIK3CA expression, PIK3CA mTOR pathway in prostate cancer, and also the MET, and how the patients there was no increment in survival even though the bony mets disappeared. Mm-hmm. So the question here is, if we think that this was because of alternate pathways that may have developed while we were trying to target one pathway, isn't it that we are realizing now that it's time for multi-pathway attack at one time? And could that be one of the reasons why PIK3CA inhibitors have not really gone a long way in prostate cancers, and we don't see them in terms of approval, or even though this is a predominant pathway? So your thoughts?
I completely agree. I wish you would be one of the reviewers of my paper, but no, I think that is a very, very good point. And I think that, do I think that just the PI3 kinase inhibitor would work? I don't think so. I think it is a combination of mTOR and PI3 kinase pathways. And I think those dual inhibitors are in development currently. And the second point is, I think that it is quite important to look at multiple lesions to see what is active. And they may not even be active as single agents. You absolutely need to combine them to be able to get the maximal benefit. And I think that this is taking into account that the tumor microenvironment is not a silent partner here. It is pretty active in maintaining, this whatever is going on in the different tumor deposits, and you cannot lose sight of it. And I think that is what the data shows.
Great thoughts. I have a question from Dr. Mandal Kulkarni again. He says, do the CTCs targeting different metastatic sites harbor the same genomic proteomic profile, or would the bone targeted CTCs harbor a different profile than that of CTCs targeting liver or lung? I think it's a great question.
So it's a question that makes a lot of sense. I think that, the situation in the mouse is much more controlled. So we start with the P10 deleted model, which is very common in prostate cancer, right? So we had 2 P10 deleted models, and we are looking at all the cells that are coming out within about how many, about 10 weeks. So there isn't enough time for these massive genomic transitions to happen. And these are all isogenic cells that have actually sort of basically landed at different regions and are kind of evolving based on their tumor microenvironment in an epigenetic way, probably to regulate their signaling pathways, right? Now, is that the situation in the human? I don't know, because I think in the humans, you seed, and then, your tumor microenvironment is going to have an effect on how you evolve in the evolution of the mutational landscape. So that probably is something that we are going to have to address to see whether there are predominant mutations that are associated with the different tumor deposits that are present at the different sites. I don't know the answer at this point, but good points.
Thank you.
Thank you very much. And I could still see some of the queries in the chat box. I would appreciate if you could please answer some of those queries.
Yes, I can do that. Thanks.
Thank you very much for your time. And we look forward for the panel discussion. Just an announcement that we are going to delay the panel discussion for next 15 minutes. We have another speaker, Dr. Andreas Moller, who is going to speak for the next 15 minutes. So can you put the slide of Andreas? Thank you very much, Dr. Shamla. Thanks a lot. Yeah. So thank you, Andreas, and I think it's a pleasure. We have heard a lot about the CTCs and ctDNA, and I thought it completely makes sense to bring you in the discussion and giving a fresh thought that when we talk about the liquid biopsy, it's not only about the CTC, ctDNA, it would be exosome. It could, and this is something I could see the title of your talk. So let me just briefly introduce you to the audience. Dr. Andreas Moller, he's associate professor at QIMR. Andreas Moller leads to the tumor microenvironment lab, which focus on advancing our understanding of the communication between cancer cells and their environment. His lab was first to show that this communication of cancer cells initiated by low oxygen environment within the tumor, that is something hypoxia, caused the formation of pervasive environment in distant organ. Currently, Moller and his lab is delineating the communication between the malignant cells and normal, especially immune cells at metastatic site. It's my great pleasure, Andreas, and please, we're looking forward for your talk. Thank you very much.
Thank you, Prashant. And let me try to share my slides again. Thank you, Prashant, Mani, and Sevanti for putting this very exciting conference together and allowing me to give a quick update on what we are doing. And I'm very excited of hearing all these great talks so far. So as Prashant just said, we are working on extracellular vesicles and using them as a liquid biomarker in cancer. So I don't have to introduce the liquid biopsy here, just to say that we are working on blood exclusively at the moment for this project, but we are looking at a few of the other body fluids as well for biomarkers. And we heard a lot about the circulating tumor cells, the ctDNA, some mentioning of proteins and RNAs. And as the last speaker, I put extracellular vesicles last. I'm pretty sure in one of the next meetings, these will move up in the hierarchy. So extracellular vesicles, very basically, we can divide in at least 3 different subgroups, which are really classified based on their size and biological origin. One would be the exosomes, the smallest of them all, microvesicles and apoptotic bodies. And I will talk on the small extracellular vesicles, or exosomes as they are called generally. So maybe most of you will know, but exosomes are really formed in the cell via multivesicular bodies. These multivesicular bodies are then secreted into the intracellular space and can be taken up by blood, lymph, or saliva, or anything like this, and be carried. Now, an exosome is really a lipid, a small lipid, the size of a virus, and we got actually into problems before because it's the size of the virus. It has a virus-like activity, and this was before Corona. So we are very silent at the moment that we work on virus-sized particles. But really, these lipids are having a few proteins, transmembrane proteins, for example, tetraspanins, which are there because of the biological origin and have protein content, which is also based on the biological origin of the cells. Now, we discussed in a recent review some of the work which is emerging, that if you, for example, have 2 different cancer cells here in blue and in gray, and you analyze the exosomes of these cells, that obviously the normal constituents are the same, but proteins, RNAs, and DNAs, as well as lipids, can be different. So you can find, for example, an intracellular protein B in cells from a very specific oncogenic mutation pattern, and a protein A from a very specific mutation pattern here. So we published some of the early work in this area and been confirmed that this is happening. And this has then obviously impacts on where these cells are going, these exosomes are going. Now, I'm focusing today on one part of our biomarker work. This is the time of diagnosis, but another clinical problem we are really working with is the outcome and best therapy, really what a few of the previous speakers have mentioned. How can we do this better? And we have an exosome-based prognosticator for non-small cell lung cancer, so how to predict whether these people benefit from chemoradiation therapy and or checkpoint therapy. This paper should hopefully be coming out soon, but I wanted to share some preliminary work today on the time of diagnosis. So if we have, for example, this patient here, it's one of our non-small cell lung cancer patients, advanced stage. And as for most cancer types in advanced stage, is mostly poor outcome. Only a few of these patients have a good outcome. However, if we could somehow diagnose this patient earlier, so in the past, where the same patient has maybe only very small lesions, most of these patients then have a better outcome, a long-term survival, or even a cure. Whereas fewer patients will have a poor outcome, a short progression-free survival and mortality. So coming back to this idea, can we actually use this kind of principle of different content of exosomes to help us with these diagnoses of cancer? So what we actually thought is, if the exosomal content is specific to the cell of origin, could oncogenic mutations in the cell be causing changes in the exosomal content? So really, a normal cell versus a cancer cell, is there any difference in the exosomal content? So what we've done is a few lines, and I present you one. We have used untransformed lung epithelial cells, HBACs, and isogenic lines of these, which are transformed, for example, with oncogenes, p53, KRAS, anything like these. We collected the exosomes of both of these cells, and we've done microRNA profiles, but also proteomic profiles. I will only talk about the proteomic profiles today. So we analyzed these Now, if we analyze these pair for here, you see we have a lot of proteins, in total over 50 proteins, which are different, which are specific to these kind of exosomes in the transformed cell-derived exosomes. We have done this with several pairs of untransformed and transformed cell lines, and we came up with a short list of 4 proteins here, which commercial ELISAs were available for. And you see, if we compare these, we have a much higher abundance of these proteins in exosomes derived from the oncogenically transformed cells than in the untransformed cells. Now, this is fine that this is working. This is a positive control for our work, but we actually wanted to transfer these. So we used a number of different cell line derived exosomes from lung cancer, glioblastoma, colorectal cancer, breast cancer, prostate cancer, melanoma, esophageal cancer, and ovarian cancer. And you can see here, if we look at these abundance of these 4 proteins, it's always much higher in the exosomes derived from a cancer cell, and we normalized it to a number of normal untransformed cell types, cell type derived exosomes, suggesting that at least these 4 markers might be interesting as they are higher in abundance in cancer exosomes. So how to translate these? And we really wanted to do something which is personalized, so which can help each individual patient. Now, the next few slides will be very busy, so I take one of the examples out, how we actually transfer these to the clinic. So here we have recruited 250 odd healthy individuals, age and sex matched, to our cancer cohort, which is over 850 patients. And you can see here for one of these proteins, the abundances of the protein in exosomes derived from all the different cancer types. So this is lung cancer, again, brain cancer, colorectal cancer, melanoma, gastric cancer, esophageal cancer. We have only 12 prostate cancer samples, 6 with high, 6 with low Gleason, and only a small number of small cell lung cancer samples. But whereas these others are reasonably powered. As you can see, for each individual cancer or cancer type, you see a higher abundance of this protein. But again, we have quite sometimes high readings in the healthy individuals. These were Red Cross blood donors, so anonymous. We can't go back. A few of them we suspect, and they're in an age bracket that they might have cancer, but discussing this maybe later on. But you can see that, for example, this non-small cell lung cancer patient down here has a lower value of VCAN in this case than most of the healthy individuals. So we actually explored if we combine all 4 markers for these 250 healthy and 850 odd cancer patients. And you see that there's a very nice mixed variety there. So we went to our biostatistician department, who wrote a nice algorithm. We can actually differentiate cancer from healthy individuals with an accuracy of 96%, with a sensitivity of 87% and specificity of 94%. Now, most importantly, or most interestingly, is if we actually take the values, can we differentiate between the different cancer types? The answer is well, mostly. So if we take a fixed specificity of 95%, you see that we can identify colorectal cancer, glioblastoma to some extent, melanoma, gastric cancer, and esophageal cancer very well from all the other cancer types. Prostate and small cell lung cancer had very low numbers, so this is a little bit biased. And non-small cell lung cancer is okay, but not really great for reasons we are not 100% sure of why these 4 proteins are not working brilliantly there. But if we actually ask the more important question for an early diagnostic is this working on early cancer patients? So only 3 of these cohorts were sufficiently powered with stage 1 cancer patients. And you can see that if we separate these into stage 1 or stage 2 to 4 patients, the accuracy or the sensitivity of detecting cancer is the same between stage 1 and stage 2 to 4, meaning that our exosomal motor protein content diagnoses early stage cancer as well as it does diagnose the whole cancer. And this might be a difference to the circulating tumor cells, or something where the abundance might be correlated more to the abundance of or the size of the tumor. So what I said so far is that we generated a clinically validated pan-cancer diagnostic biomarker based on the exosomal content changes. So how can we utilize these in a proper clinical setting, not in these retrospective cohorts? So for this, we are working here in a clinical trial with David Fielding at our hospital, who is seeing a lot of, he's in a thoracic ward, and he sees the patients which get a suspicious X-ray or anything, ultrasound, anything which are normally referred by the general practitioner to the hospital for follow-up. So these patients come to the hospital, lots of tests will be done, biopsies, PET, CT, MRI. So these patients are potentially having lung cancer. So all these tests will be done, and then there will be a diagnosis formed on these biopsies, the classic biopsies and imaging. In our trial at the moment, it's around 60% of these cases are cancer, and pretty much all of them are non-small cell lung cancer. We have a couple of odd mesotheliomas and small cell lung cancer in there. Now, the important thing is that we actually get blood samples at the beginning when these patients come in, and none of these patients is healthy. So all of them have something, mostly benign inflammation-associated shadows on the X-ray. We get the blood samples, and until we actually know the outcome, it's a few weeks. We have done the ELISA-based testing on these samples, and we have a very, very good data, but I want to actually skip because I prepared these in response to really what Klaus said before, that we should really try to validate our data from independent cohorts or independent sources. And while we haven't done this completely independent, we actually have started a collaboration with a group at the University of Queensland, where they are using nanoshearing devices. So we actually take the blood sample. We have different antibodies to capture exosomes. And here, for example, we have exosomes with which are positive for our markers. Here only 3 from our 4. Then we can actually have a SIRS sandwich assay with a multiplexed readout, means we have SIRS probes which are detecting the abundance or the position of these different exosomes. So if we use this for CT96381 and tetraspanins for exosomes, you can see we get these multicolor readouts, which are indicating the abundance of these proteins. But what we have done in the first trial, we have actually taken 18 samples, given to them blinded because we don't even know what it is. And we have here the image for our signature proteins, as we call them, the 4 proteins for benign cases, and down here for 5 malignant cases. And you can see just visually that there's much more of this protein on the exosomes here than in the benign cases. So here's the quantification. And again, it turned out that we can accurately predict 9 out of 9 malignant cases and 7 out of 9 benign cases with this approach. So these were the first 18 cases. We have now around 80 cases collected from this cohort, and we are progressively putting them through this test in a really double-blinded fashion, validating really that our assay might pick up cancer exosomes at a very early stage. So all the patients I have to add are stage 1 or 2A patients, which are going through the trial there. So in summary, we have, as I've shown you in this short talk, generated a clinically validated pan-cancer diagnostic biomarker, which is really based on protein changes induced by oncogenes in the cancer cells. And this exosome biomarker can be applied to prospectively collected and outcome-blinded patient cohorts, and we are able to differentiate benign and malignant cases. So one of the applications, for example, lung cancer screening could be, if we are 100% or very confident that the patient is benign, we could actually reduce the workload for our clinical colleagues in the thoracic unit to screen all the patients with quite labor and money intense pathology and imaging approaches. So we are I showed you before, we have a lot of different biomarkers. The problem was that very few were available for ELISA testing. A lot of the other proteins, there are no ELISAs available, but now with the SIRS platform, and we have a different platform as well, which are independent of commercially available markers or ELISAs, we can actually evaluate additional proteins and will filter back a few of the patient samples into these cohorts. Now, I would like to thank a lot of my collaborators, a lot of the clinicians who kindly provided a lot of the patient samples. It's quite hard to find blood samples and correlated patient data, so I'm very grateful for all my colleagues, Matt and his group, who are doing the SIRS detection work, and the patient and the families and our funding sources. But I would really like to highlight the work my tiny group has done. So we have isolated for this project alone over 2,000 patient plasma samples, and one of these robots here, like Richard and Kekolani, Luis, Edna, Senyong, and Erica, and this picture, a couple of people missing who joined a little bit later. They can maybe isolate 32 samples per day. It's a boring all-day job, so they have done great work in doing this and driving these projects. Thank you very much.
Thank you very much, Andreas. Brilliant work. And this is where I would say that there's a transition from bench to bed. Amazing data. I have never thought the way you have explored the exosome biology. Two quick questions. And the first, maybe I missed it. Did you multiplex your biomarker to reach the highest level of sensitivity and specificity?
Yeah, so that's what I wanted to point out. If when I showed you this VCAN alone, it is overall, if you look at the patient populations and healthy subject populations, it is predictive and it has a very good area under the curve. But to tell the next person going through the door what is happening, it's not good enough. We needed to complex the data from all 4 samples, and only the 4 samples were predictive for on an individual level, so not overall for hundreds of patients, but we could actually really predict for an individual patient with 97% accuracy whether this is cancer or not. Maybe I want to give you 1 second there. As I said, one of the caveats is who is healthy. Especially the average age of the cohorts is in their mid-late 60s or mid-60s. And we have a couple of blood donors which are in their 90s, a few people in their 80s. Now, especially for a man, you would expect that a lot of them have undiagnosed prostate cancers, we heard before, and so on. So we have a few of the cases which have very high ratings, and we actually asked our biostatistician to say what it is, what they would predict if this would be cancer, and they would actually come back, and a couple of them came back with positive for colorectal cancer, a couple of them came back with, or quite a few for breast and prostate cancer. We can't go back, but that's where healthy is a little bit subjective, I think.
So Andreas, you always come up with some surprise, I would say. So any plan to see the correlation between exome signature and CTCs, or?
So look, when you asked me whether I want to join, I said, well, only if I can surprise you. So I'm always happy to surprise you. Now, look, for this part, it is really, reasonably brand new. So we have a paper which is going through the different co-authors, which we hope to submit early mid-February now. But we have a prognostication of outcome in non-small cell lung cancer, and there we have a clinical trial which was meant to start recruiting last year, but because of a certain corona situation, we were not able to really get it started, where we have a couple of local collaborators who work on ctDNA. One works on circulating tumor cells. Another person has a microvesicle signature and our exosome signature. So we want to combine these and see, is there an additive synergistic effect? Are they redundant? So do we always pick up the same signal as someone else? And maybe the other people can comment, but I'm not aware of a bigger kind of trial where has been tried, or if so, I'm more than happy to see how we can contribute and test our samples.
Thank you very much. Thanks a lot, Andreas. So we are now going to very close to the meeting, the last session of the meeting where we put the panel discussion, and I will just ask Sevanti to please take the lead.
So I just need to, so actually Prashant is moderating the session. I'm just introducing the panelists. So Ashish, could we please have the introduction slide? So we've run late. I don't think, Prashant, you've seen the chat box. Dr. Klaus Pantel had to leave. So Klaus we'll be missing Klaus in the panel discussion.
Okay. Mm-hmm. Okay, so may I request Rick to join the panel discussion?
Hi, Prashant. Well, I'm happy to I'm happy to be here. Yeah.
For you, Rick.
Thank you very much, Rick. Thanks a lot.
Thank you. I'm happy to participate at whatever I can. Thank you.
You stayed up late anyway.
Couldn't tear myself away. It's been really great. Thank you.
That's amazing. So it's me, it's Mani, it's Dr. Shamla Maheshwaran, it's Dr. Pastoon Kazi, it's Dr. Govind Babu sir, Dr. Hima Bindu Gadipati. We're just meeting her right now. Dr. Hima Bindu Gadipati is the chairman and managing director of VCR Park in Visakhapatnam, India. And she's a leader in oncology and in translational work. She's a thought leader in terms of entrepreneurial approach to bringing translational oncology to global forefront. And we welcome her here in the panel today. And of course, we heard Dr. Andreas Moller with an amazing talk. And we welcome all of you all for this panel discussion. And our good friend Prashant is going to lead the panel discussion. He's moderating the session. We're really going to miss Dr. Klaus Pantel, but I hope we're going to have an engaging discussion. And in the middle of the night, and truly, if Dr. T. Raja is listening from last night, this is turning out to be a midnight oncology to try and connect, two continents, actually, several continents, and bring it all together. The floor is yours, Prashant.
Okay, thank you, Dr. Sevanti. So I'll just share the slide.
Prashant, as you're doing that, disclaimer for me, no tough questions.
So I'm getting I'm getting trouble with the sharing. Janni, you're there. Can you share the slide? Seventy.
Yeah. Yes.
So I sent you the slides. Would you mind sharing? I'm getting trouble here.
One second.
So I think this is the toughest job I got it, to sit in the chair with the elite speakers, leaders, experts. And I had a very hard time thinking that what is something I should ask. And also the other way I was thinking that this is the best opportunity for me to get my queries done and get the views of the world leader experts. So I really look forward to your comments, suggestions, and opinion.
I'm happy to go first. I thought that, you guys had done a terrific job. I think I have to be honest, because I think sometimes because of time constraints, I came into this thing thinking that I would actually sit for half the talks and then go and do what I had to do. But I actually, listening to all the talks, I rearranged most of my commitments so that I could actually stay for the full length. So I think that it was tremendous. And I was honestly so proud to be a part of it. And I just did, that was not lip service. I really meant it. I think all the talks were tremendous. And I think that there was so much thought that went into the work that each person is doing. So I really enjoyed it. And congratulations and kudos to the three of you who actually put it together.
A lot of credit goes to Prashant, actually. I think he's done he deserves most of the credit.
Thank you very much.
Well
Thank
done.
you for the generous word. Appreciate it.
And while I'm just putting up the slides, I think Dr. T. Raja is also with us. And Prashant, I would like to invite him to the panel as well.
Please, please.
Sir, are you there?
He said he's on a prowl.
So couldn't leave you alone for the midnight oncology discussions.
No, he sent a message. He's on prowl.
Oh,
So I said the lions are on the prowl.
Yeah, you're the lion.
I said lions plural, Govind.
Thank you.
But truly, this turned out to be a great meeting.
Yeah, I agree. Totally agree.
Initially, I thought, okay, liquid biopsy will hear a couple of talks, but I think this is mind-boggling.
Correct.
This is beyond the current, I would say.
Yeah, true. And Raja, frankly, a lot of it was Greek and Latin for me. But it's good to know, at least I could recognize it was Greek and Latin.
This is the goal, right? The most important thing is clinicians and scientists talking to one another is the most important thing. I think this meeting...
I agree, Mani, and I must thank Prashant for that.
Yeah.
Thank you.
Prashant, all yours. You just tell me next and I'll do it now. I'll know a little bit of your slides ahead of time.
Thank you very much. So the highlight, I just kept it liquid biopsy for precision oncology. So could you please move? Yeah. So just to the audience that when we talk about the liquid biopsy, we should not forget the tissue biopsy, because we learned a lot and still it is in practice. We use tissue biopsy and I hope over the time things will change. And when we talk about the liquid biopsy, yes, cell-free DNA is something which we have explored in decades. CTCs, and you have heard from the world leader, there are challenges with the CTCs, with the enumeration, numbers, the biology, but there's certain advantage what we get it from the CTCs, which may not from the ctDNA. Exosome, extracellular vesicle, what Andrea just now talked, and this again fascinating that we could use other liquid source And I would say that again, saliva for the head and neck cancer, I think that is another source. And many more, tears could be. So something where the liquid biopsy, when we're talking, it's a multiple facet. And I think we are really ready to go and understand that how it will make our life easy. So let's go to the second slide.
One quick comment on this slide, if I may.
Please.
I was going to say this question comes up all the time, and I think there's always criticism and discussions regarding the question of liquid versus tissue, or even within liquid, ctDNA versus CTCs. I don't necessarily see it as an either or question. Where tissue doesn't facilitate or is not available, liquid is complementary in that scenario. And then within liquid too, as we saw in Dr. Pentel's and other presentations too, that CTCs have their own role to play. If you're talking about proteomics or RNA, you cannot derive that information from ctDNA, and CTC has its own fallacies too. So they're all complementary. I don't think it's an either or question. It's more of a, it's not going to replace the tissue question. I don't think that's even needed. Tissue will always have its own role, and liquid will have a complementary value from a clinical standpoint. And within CTCs and ctDNA question too, I don't think it's an either or question. I think they're complementary to each other.
I have actually, since this is a panel discussion, I have a question to all of you who have actually sort of such extensive experience looking at ctDNA. I have actually talked to a few people who say that, finding amplifications using this ctDNA analysis is not very reliable. Is that your experience as well when you have actually used it for clinical purposes?
I think one big anecdotal experience is the fact that the folks who may say that amplifications are not reliably detected, the biggest question I often ask them is, when did you check the assay? With liquid biopsy, the problem is not just the sensitivity of the assay, it's also about the timing of the assay. If you check, for amplification in somebody who's responding to therapy, as we saw, ctDNA may be, not detectable or very low, and then that's when you miss the amplifications. If you truly, test the liquid biopsies at baseline in an untreated patient, just like you would do with tissue at the time of diagnosis, some of the data and experience that we have with HER2 amplifications, for example, the group from Guardant Health, they published their data in one of the AACR journals in the last few months. The sensitivity is above 95, 96% in that scenario. So the biggest question often comes up is, one of the experts from MD Anderson has a nice terminology of how to read liquid biopsies. He calls it, Dr. Kopetz calls it the positive report. And then you have within negative, you have your uninformed negative and your informative negative report. And what that means is, if you barely see any mutations at all, in that scenario, you cannot say the person is HER2 negative. The report itself is bland because you don't have enough substrate for analysis. And your informative negative is when you have a ctDNA, which is over 50%, 30%. Some of these GI tumors are very high shedding tumors. So in that patient, if you don't see an AMP, then you can reliably say that this is an informative negative report and ctDNA is truly negative. So in fact, some of the new trials, we're opening a trial, second line HER2. FDA actually is accepting, they wanted the liquid biopsy as the amplification companion test of choice, not tissue. So I think it's more of a timing issue and not necessarily a sensitivity issue, at least for some of the clinically relevant amplifications.
Pashtun, can I briefly come back and to your first point about that the pathology or the tissue biopsies might not become completely redundant and it's a complement to do the liquid biopsy. I think this is correct in most cases. However, if you have situations, for example, patients with or individuals presenting with a low risk of disease or anything like this, where a biopsy might have quite significant comorbidities as a consequence or a high chance of it, you might find, and I know there's discussion at least here in the Australia, New Zealand region going on, what kind of tissue biopsy can be not replaced, but maybe preempted with a liquid biopsy if you have already a low chance of an individual having a certain disease and your liquid biopsy says, yes, it is a very low chance, clinicians are discussing that they might be, happy to then forego a tissue biopsy. So I think, I agree, and I think the best is that we have more tools in the toolbox to actually look at disease, and especially after the primary tumor is gone, pretty much never a tissue biopsy is repeated for a metastatic site unless it's a trial setting. So I think there the liquid biopsy really will be very important.
Yeah, I can give a very true life situation. We had a couple of patients, as is a tertiary cancer referral hospital. So many of these patients have a diagnosis done with a fine needle aspiration. We had some patients who had a FNAC done of a lung lesion and was reported as lung carcinoma and adenocarcinoma, but the patient was too sick for a repeat biopsy. So, there we did a liquid biopsy, we did mutation analysis for EGFR. Fortunately, we did find in 4 of these patients EGFR mutation. So we were able to treat these patients based just on that. So there are clinical situations where we may have just a needle aspiration for diagnosis, but no more tissue for mutation analysis. And then we have done with ctDNA.
And also there are some situations where biopsy still is not recommended. So, some of the, cholangiocarcinoma, there is still that concern that you may see the tumor. So a biopsy actually is indicated in some of the protocols. So in that scenario, often the question comes up is, are you comfortable in treating the patient without tissue? Well, I don't necessarily have to be in that situation anymore because I could rely on a liquid biopsy in that scenario too.
Right. Again, for hepatocellular carcinomas, this is another issue, yes.
Me and Prashant are chatting on the side. I'm telling Prashant, you're not needed. It's a passionate group of people. They're happy talking. You can just leave them alone.
Yes, Prashant, go ahead. Sorry.
Please.
Go ahead.
Actually, could I ask a question on that same topic just quickly? Because, we used to always say, and now I believe you have a chance to really know that you have a benefit from that integrated sampling, particularly in advanced stage disease, that you don't have to find every met and biopsy every met. It's all coming into the bloodstream. Particularly, is that actually panning out like that you see? We do know about clonal evolution at different sites. Are we sort of seeing that we're covering that without actually having to sample all those sites?
Yes, many times it's happening, yes.
And we recently published on our experience in these FGFR fusion cholangiocarcinomas who get exposed to FGFR fusion inhibitors. And on the resistant mechanism side of things, they actually acquire many gatekeeper mutations. So on the liquid, you can pick all of them. And some of these patients who've had tissue biopsies, sometimes in the biopsy, you don't even see a single clone, or you might see one of the several clones on liquid. And same goes for rectal as well. So indeed, it is of value in these patients getting exposed to targeted therapies.
Thank you. That's fantastic. Thank you.
Prashant, take over quickly.
No, it's great. If you wait, we will continue.
Yes.
So I'll try to reach the leaders. So so the next question which comes in my mind, the potential of liquid biopsy for prognostic, predictive, and early cancer detection. And this is what we are talking. So promoting timely, this is something I would say that, do you think this is important? Second, targeted intervention via liquid biopsy techniques. So can I get the comment from Dr. Sevanti?
So my whole approach has been doing longitudinal liquid biopsies to understand the targets and to modulate my treatment accordingly. Many a times there are multiple alterations that are that show up, and that becomes a challenge. Just as of yesterday, there's a case that we discussed, pheloids breast cancer, where we saw PIK3CA as well as EGFR mutation, and there's no data. So yes, there is for PIK3CA, but there isn't enough for EGFR. And which one do you target? There's always a challenge with that. And in that situation, I'd ask this question also to Dr. Kasi, and you go ahead with targeting what is most understood. And even with that, this patient did not respond. So what was the interplay? One doesn't know. And that's where the concept of multi-targeting comes in. And sometimes if the patient is able to handle a combination, you can see disease stabilization and even regression and treatment response. So I do utilize ctDNA alteration, understanding of ctDNA alterations for strategizing my treatments for patients.
Okay. So what you could see this graph, and it shows the clonal evolution. A patient post-treatment, they were enriched with the P53 mutation. Over the time, you could see some other mutations enriched. So can we consider delivering adjuvant treatment to patients with minimal residual disease, given their higher risk of recurrence? And I will go to Dr. Kasi. What's your comment?
This was the most talked about question, last week from the ASCO GI conference. As of right now, the active trials, there is the Bespoke study that we're leading, there is the COBRA study with an RG trial. That's exactly what they're trying to answer. Is it just a prognostic marker of recurrence, or can you truly clear these, minimal residual disease by adjuvant therapy? Or the other question is this, stage 4 disease that actually hasn't macroscopically, radiographically recurred as well? So we don't know. Like earlier, there was a discussion regarding, the breast cancer staging system being revised. There's discussions in the colorectal groups of experts too. Somebody who is NED but is ctDNA positive, is that a different stage system altogether? Somebody who's stage 2 but ctDNA positive is 100% risk of recurrence. So, theoretically, I would say yes, but practically speaking, we will know the answer with the ongoing accrual to the active trials.
The data coming is really exciting, but I will play the devil's advocate. For carcinoma ovary, we've always been following up with CA125 as a marker. But, we have had conflicting reports whether to treat or not. So today's consensus is that don't treat elevated CA125, follow them up till a tumor is picked up on an image. So I'm just playing the devil's advocate here.
So this is exactly what I was trying to say. Field is still stuck. It hasn't moved with the, so for us, for Dr. Govind Babu sir, myself, those of us who don't have clinical trials, we are still at crossroads. We still wait for that appearance of that new lesion on the scan. And that's where I feel it's really getting delayed for us. And even as we know that this is a patient who's becoming stage 4, what is the best treatment? We don't know that. So yes, we know this is doing poorly, but is the next treatment the best treatment?
Yeah. But again, Prashant, I would give you a fantastic analogy that we follow for treatment today is chronic myeloid leukemia. We follow BCR-ABL quantitatively and we treat them. So yes. I'm convinced that this is going to happen in solid tumors, if not today, tomorrow. So I think that is the best analogy we have today.
Dr. Prashant.
Dr. Raja, please come in, please.
See, I think what Govind pointed out is he played both for and against at the same time. Listening. So one is that CA125 not acting on it is a different clinical decision, but the fact remains that if the CA125 goes up, and definitely it's a harbinger of a relapse. When do we intervene is a different issue. Same thing goes for PSA. If it, if you talk to the urologists who have done radical prostatectomy, if a PSA level is even from 0.5 to 1 or 2, it goes up well within the normal limits, then they consider acting on it, adding local radiation and all that,
right.
and so forth. Now, answering your question on MRD itself, it is well established practice that treatment decisions are made based on MRD in acute leukemias. Acute lymphoblastic leukemia is the norm, how we select our transplants in ALL in children and also in adults. And in AML also, we now started routinely doing our MRD. Now we have recently added myelomas to our portfolio. Yes. It can be called as a liquid tumor and a solid tumor, but myelomas have already, we now routinely do flow cytometry too. Basically, they are all like establishing the signature of the disease in the lowest detectable form. The only question is how reliable, sensitive, and specific is the issue. But we are very aware of the work happening in the colon cancer, where colon cancer post surgery or post, we are well, follow up emergence of either CEA or any of these circulating tumor cell is definitely considered as an harbinger of relapse. It's already established. Similarly, I think it's a TIC-TAC study in prostate cancer. TACTIC study in prostate cancer, once again, longitudinally follows. So there has been enough. The only trouble is we do not know when to act on, but everybody knows that something is going to come up. That message is very clear.
Yeah, yes, Raja. In fact, yesterday I showed a paper from Nature Medicine just published, I think, in January 2020. Now they're talking of MRD in breast cancer. So I think it is going to come up.
And one small point I wanted to echo is it depends on the disease context and what you have available. So right now, for example, a classic situation where ctDNA MRD you would act in colorectal cancer is it's a stage 2 cancer patient where you were initially thinking of not giving chemotherapy.
Right.
If the MRD is positive, well, that's a black and white question in my mind that yes, that person should get adjuvant chemotherapy, whether it's 3 months, 6 months, and then within colorectal 2, there is now a spectrum of intensification from surveillance to 3 months of capecitabine to FOLFOX to CAPOX to 3 months to 6 months. You could potentially intensify or de-intensify the treatment based on this as a prognostic marker. So I think precision medicine is testing is half the battle. The other battle is the drug itself, and now with immunotherapy for MSI high, you have other more actionable targets. I think with ovarian cancer too, when the CA-125 data came out, none of these other fancy drugs or target therapies existed.
Absolutely.
Better good early intervention in my mind would show the difference, and the answer would be disease context specific.
One other issue, if I may add to Dr. Pastoor, is that sometimes we see, but we do not know what we are seeing and what exactly is the effect of what we see. I can try to explain that. For example, we all know that the BRCA1 and 2 mutations would immediately confer a HRD deficiency. But then we also know that the HRD deficiency is conferred by several other genes. Then we understand that if we start measuring them and trying to equate the HRD score with the PALB2 or the RAD, there is some sort of a difficulty. Then I think recent papers say that it's not only demonstration of those genes, you need to demonstrate the effect of the gene. Basically, if you can demonstrate the LOH, then that should be good enough to, so basically it is not only detecting the abnormality, but if only we can detect its work, if proteomics can be added and we can demonstrate the effect of such a change, probably that will be the mutation or anything to target.
So thank you very much. I just may request the panelists, please briefly answer so that we would, in the interest of time, I just make a request and we could just go through all the slides. So going to the next slide, establishing the analytical validity of circulating tumor DNA sequencing for precision oncology. And I find two trouble. One, which is limited input material. And you know that the mutations showing variant allele frequency below 0.5%. So there would be a sensitivity issue. And the other one would be the ctDNA fragments. And my belief is that there's high probability that we could miss the mutations or we would find false negatives. So I quickly want a comment. So Dr. Kasi, could you please briefly comment both the points?
With the commercially available platforms increasing sensitivity, I don't necessarily foresee that to be an issue, at least, 2021, with at least some of the available platforms. The fragment part you bring up is an important issue, because large gene rearrangements in terms of BRCA or ATM, they can be missed, and also fusions can be missed. But when found, they are indeed specific findings. So, every test will have its challenges and limitations.
Okay, Dr. Sevanti.
So I agree. It is just, it also depends on where you're getting the testing done. And we have to be very cautious, mainly because there are lots of newer genomic initiatives happening across the globe, and validations are happening that are internally preserved. We have to be aware of the labs, and mainly in a resource limited setting like India, we have to have our knowledges intact in terms of where the testing was done and how to interpret the test results. And I think it becomes very meaningful to interpret a positive or a negative in a clinical context, as well as with the background of where the testing is coming from. And whenever there is a doubt, you need to retest.
Excellent. Can we move to the next slide? So yeah, this is something you could see that some tumors are known for cfDNA poor. And I just made a shadow here, the pink, that renal, glioblastoma, bladder, pancreatic, compared to others. So only in high burden settings, what would we do with the low burden setting? Dr. Govind Babu, could you please comment?
I think I just touched on this during my talk yesterday. So there is a new platform and a new way of doing it with a genome-wide sequencing. And they say that even cell-free DNA as low as 10 to the power of -5, you're able to get good data. So as you see, this paper is from 2018. I think we've come a long way from there. And I think we are just getting better with technology. I'm sure even with low cell-free DNA, we will be able to get good information soon.
Okay. Can we move to the next slide?
One comment, brief comment, if I can add to the previous slide. Regardless of the disease, also the sites of metastases makes a difference. The group from Japan presented data that liver was the highest shedder, followed by peritoneal, followed by lung-only metastases. So, can't take the clinical context out of the equation. So even within colorectal cancer, the sensitivity varied from 50 to 85%. So, it's hard to say not good for pancreas. It also depends on the sites of metastases, the biology. So I won't say that I won't do it for a pancreatic cancer patient. And even for colorectal, you have to interpret it in context of the sites of metastases.
And the field is evolving, and I think sites of metastases and the yield for different kinds of cancers are also turning out to be different. So this is still to be studied fully.
I just need
right. Brain metastasis, the shedding is low. So
I've been trying to resist from saying, but I just need to say, from the slide 1 onwards, commercial platform versus like in a tissue biopsy versus ctDNA. In a tissue biopsy, when you take it, you do a whole genome sequencing or total RNA sequencing, you get the whole picture of both known and unknown. But in a ctDNA, you are always trying to investigate on knowns, not on unknowns. So in other words, we are focusing on the mutations which are previously known, not on something which is not yet discovered to play a role. Therefore, even in the previous slide, while you say that there is a differential sensitivity on the known mutation, there may be differential sensitivity to things which are unknown. So which we cannot explore because we don't have a way of doing that unless otherwise we study the tissue biopsy to get a better picture of what to look in the ctDNA. Thank you. Sorry.
No, absolutely. Yes.
Please.
Next question.
Okay, so this is something very interesting, and this actually bugged me a lot. And I think Dr. Shamla is here, and this is the appropriate time for me to ask her. So CTC's number, that we are struggling, and that's also depend on the cancer types. And this is something Sales Search claim, and the number of CTCs you could see. There always a way you would find CTCs in benign disease and healthy controls. And you could see the numbers that there are chances 3% cases. You could end up with more than one CTCs And again, in benign disease, 7% numbers. And can you just move to the next slide, Dr. Sevanti?
Yes.
So, and then I'll put my question. So this is something which Klaus has done with Alex. And what they have done actually, this is a case where colorectal cancer patient, and they used two different methods. So EP spot, CTC plus, and cell search. And what they found that the percentage, the 10 out of 53 and 6 out of 53 patients were found positive. Now, when they followed them, so this is interesting, when they longitudinally followed after 3 years of the follow-up, none of these patients have been diagnosed with the colorectal cancer or any other epithelial cancer. And this is, can you just press 78? Yeah. So would I put it that there's a specificity issue? And I would very happy to hear doctor's comment. But first, Shyamala, please comment. What is your thought?
So I think that, I mean, we initially, when we actually looked at our circulating tumor cells, we relied on staining, and it's a mess. Basically, you have these dual positive cells, and you have to really very carefully set your gates to be able to find cells. And sometimes I think without molecular analysis, I sometimes wonder whether you can actually rely on just staining to make a call. And I think some people are very confident, and probably they have additional data to support what they are claiming. But I think that, you definitely need some molecular analysis to make a call. Now, having said that, there was a paper by Ben Stanger who actually said that during pancreatitis, right, that you do actually have cells shedding into the blood. And so I don't know whether, this is real or not, but I am not surprised given that they were using, the cell search primarily uses staining to actually sort of score their cells.
Excellent. Rick, your quick comment?
Yeah, look, I agree with that. I think the LE spot is a functional assay, so it's possible that those numbers are on the lower side because they're selecting for cells which are producing those cell activities. But look at this. There's a variety of data. I mean, the data that we've seen with cell search and that Massimo presented yesterday has been reproduced just so many times, and so it's potentially just it's a disease type issue, but there's I've had a lot of trouble finding in early stage disease without that sort of technology at an RNA level, trying to find meaningful data. Yeah, so meaningful data. Yeah.
Yeah,
most of the data has been, has been generated with patients already known to have cancer, right? So I think that when you find it, you have already a correlate because you have a phenotype. But I think when you talk about early stage benign ones, I don't think, you would need tremendous amount of data to make sense of what it is. But I also worry about the specificity of the assay.
Agree. Mani, your quick comment, Mani.
No, I think I second all the comments made earlier. It's again, the CTC, detection in general, like, we are looking at a tumor cells which are, shed into the circulation by two ways, right? One, you can have an active entry of CTCs to the blood. Another one is called a passive entry of CTCs to the blood. So in case of active CTC, where the tumor cells are finding a way to get out from the primary site, survive in the circulation, and try to establish a tumor in a secondary site. Those CTC, if you detect them, they will try to kind of do something at the other end of the spectrum, other end of the equation. On the other hand, there are passive CTCs, which are getting into the blood due to leaky vasculature. Now, those CTC, if you detect them, yes, you can detect them, but they are harmless. So they are not going to do anything. So as a result, in a long run, the outcome is going to be different. So unfortunately, the detection system what we use today are able to detect CTCs, but it is not able to distinguish a CTC who is an active versus passive. So therefore, I think it's difficult to extrapolate or trying to kind of see why they did not develop tumor in the later stage. I think it's difficult at this point, in my opinion.
Excellent. So this is something, again, the buffering question that I always realize that there are two communities, the people who are working on CTCs and there are people who work on ctDNA. And I have seen actually in abroad also that parallel meetings running. The researchers called to deliver a talk on CTCs and at the same time ctDNA. And this is something, you know, that what way we should go. And this is some, you can see the genomic alterations. If you see cell-free DNA versus ctDNA, CTCs, not much difference. Even though you accumulate both, you may not achieve something very superior. So my question is, Sevanti, can you please put the next, I think I'd put some, yeah. So CTC or ctDNA in liquid biopsy, which one you'll pick and why? So quickly, Dr. Maheshwaran, please make a comment.
I am in the camp that believes that all of them are important. I feel like, you know, the blood is the giving tree. So you have, you know, so many different compartments. And, you know, we currently have a project where we're actually looking at the proteins in the blood to see whether we could actually identify smokers who are likely to develop breast or lung cancers, who are, you know, so you're looking at this high risk cohort to see whether we could actually use mass spec. The sensitivity of it is improving as we speak to actually sort of identify patients who are likely to kind of move on to develop lung cancer. So I think that basically all these components are required to actually sort of make an informed decision. And for example, while ctDNA seems to be great in terms of kind of informing as to what therapies you should do, I think that, you know, CTC cultures can be extremely informative as to, you know, what kind of, in drug screens, right? Because I think these are all highly metastatic cells which are evolved. They have mutations that, you know, that you rarely come across in primary tumors. And what are those tumors doing there? Are they kind of really drivers, or are they, you know, just resistance mutations without being drivers? So I think these are all the important questions. So I am generally in favor of incorporating everything. I realize that it's not possible. It's a real tall order, but I think all of us need to kind of come together to be able to make sense of what is needed to get to that next step, which is really early detection, right? I think that's where the biggest bang for the buck is at the end. So,
yeah. Exactly. Dr. Kasi, quick comment.
I was going to say the question is invalid because, like Dr. Maheshwaran said, it depends on the clinical context. There are situations where, you know, if you're talking about immunofluorescent HER2 expression, well, ctDNA will not give you that information. Early detection, a lot of the work right now is ctDNA, and within ctDNA, there's methylated markers. So it depends on the question you're asking. Again, these are complementary methodologies, not either or.
Excellent. So I'm going to Andreas directly. So Andreas, there are multiple studies, good number of studies over the time you could see. And I collected the data till 2019, and people have used exosome for diagnosis and of course, protein-based diagnosis. What I found that prostate cancer and glioblastoma mostly used. So my question for you, current challenges for EVs studies using blood sample and applications. And the second, the extracellular vesicle as a platform for liquid biopsy in glioblastoma patients.
Okay, I think, look, I can't say I read all these hundreds of studies, but I read a fair share of them. And I think there's a very big problem in the field, or I think we're addressing it, is the reproducibility of the data. There are lots of different, I think it's similar to what I've seen other people go through in the CTC field a few years ago, that we have different methodologies of isolation, a different definition, what are exosomes. So if I would be in a talk in our society, I would be slaughtered for using exosomes because they come up with new ways to actually define the different vesicle subpopulations. And we are just really understanding the complexity. The problem is some of these work, especially the 2016, 17, 18 time, there was a lot of contamination. We have a lot of proteins in very popular, data sets in the mass spec, proteomic profiles, and in online data sets, which are clearly contamination, which are not exosomal proteins. A lot what has been done is single protein, and there are a couple of very, very prominent studies. One protein which can predict the presence of a certain cancer or the outcome or anything like this. And it turned out that it's a lot of imagination and it's just not reproducible. So I think that's a problem in our more advanced study of the prognostication of lung cancer. We have actually gone beyond what we should have maybe done, but we actually sent our ELISAs, just the antibodies labeled with 1, 2, 3, 4, 5 to international groups and said, you have your groups, your cohorts. Here's our SOP for the exosome isolation. Can you please run it and see if you see something or anything? And I think this is what is needed in the EV field, the same, I think, as the ctDNA, CTC field and others. To your glioblastoma, I think, look, we also have glioblastomas in our studies. I found it very surprising, and it was my reaction to reading some of the studies, because I always thought the blood-brain barrier would prevent or reduce the number of EVs from glioblastoma in the blood, but it turns out that they are a lot of glioblastoma EVs in the blood to be found. So it's a very low-hanging fruit. The only problem I have is really, even if you detect glioblastoma early, so what? I mean, there's very little long-term benefit or curative therapies there in most of the cases. So GBM especially. So and that's what a couple of these studies have shown. So I think we need to move beyond the pure diagnosis, and that's a little bit what a few of us in the field are thinking, that if we know what the cancer is sending out, we might actually use the EV to target the cancer, and specifically, so there are different ways we could maybe do this, but it's a technological challenge which the clinicians and basic EV researchers can't do. So we need a lot more chemists and nano engineers and everything on board to help us with this.
Excellent. Can we move to the next slide? So this is something, what next for the liquid biopsy in precision oncology? We can skip that slide. Okay. So could liquid biopsy be more cost effective, and current cost obstacles and running cost of NGS is a bottleneck? And I'll just go to Dr. Hima Bindu for her comment. Please unmute your mic, Dr. Hima Bindu.
Yeah, thanks for that. I just wanted to thank Dr. Prashant and Dr. Sevanthi for inviting me to this eminent panel. It truly has been a pleasure and honor to participate today. And I wanted to say that I really learned an extraordinary amount in the last few hours. So kudos to you for putting this together. And thanks for inviting me as well. So I just wanted to share my perspective on a couple of different aspects, which kind of lead up to the questions that you're asking here. When we think about translation of this evolving technologies in the CTC space, it's really been a tremendous evolution over the past decade, especially. I know it's, there's been over 2 decades of work which has gone into enabling where we are today, but really in the last decade, you've seen an exponential acceleration on a number of different fronts. One is really, in terms of the regulatory pathway. And this is of key importance to me because, as a number of you know, I was part of the US FDA Division of Oncology. So, it's really of interest to see, in terms of the regulatory pathway, how this has been facilitating and removing some of the obstacles to broader implementation, especially within the community. In 2013, the cell search platform was approved by the FDA, and at that time there was a lot of ambiguity about, what are the mechanisms that we're going to use to facilitate approval. More recently, I think with the breakthrough designations that both the Guardant as well as the Foundation One platforms have received, there's been a lot more clarity. There's been a lot more thought with respect to finding more coherent mechanisms to facilitate adoption and approval. And recently they were both approved in August of 2020. And this has been a warm welcome. And I really think that it's one of those key milestones which will enable broader adoption because it enables a viable mechanism to facilitate reimbursement for treatments for patients with non-small cell lung cancer as well as prostate cancer for both these assays. So they went through the mechanism of companion diagnostics, and Medicare as well as a lot of the private insurers were very comfortable with that. So this also provided the ability for physicians at the community level to, first of all, feel that, okay, this has been validated. They feel much more secure in offering this as an option for their patients. And number two, it also provided the opportunity to remove that key bottleneck of how will we pay for this and who's going to reimburse for this. Now, this is obviously in the developing countries, much more of an issue still, and there's still miles to go. I'm sure Sevanthi can give her input about that, but these are not cheap, especially given the context that, you cannot complete, they're complementary to tissue biopsies. They're not mutually exclusive, as has been pointed out by a number of you over the last couple of hours. So in that context, if this is an additive, if it's a complementary, then you're adding to the cost. So how do you justify it, from a monetary perspective? So, I think that this is a very lengthy and complex discussion, which this probably wouldn't be the right time to go deep into it. But, I think that there are some advancements, especially in the regulatory pathway, et cetera, which will help facilitate this. But then, there's a lot more mechanics surrounding that, which have to be fully evaluated. And the second thing, I just want to point out is oftentimes it's not really what we think, which are the key bottlenecks. I believe that, a lot of it will be determined by the community oncologists themselves. Remember, they're being hit with this avalanche of information. They go to ASCO and, it's not just 2 or 3 tests. Now there's so many other platforms on the horizon. And, it really overwhelms them. They don't have the time or the space to really grasp the nuances. So sometimes, they're very resistant to adopting a lot of these testings, especially if you tell them, again, that this is complementary. It's not going to replace tissue biopsy. Unless, as you was rightly pointed out, in some cases where it's just impossible to obtain those samples in those patients with hard to get lung tumors or, cholangiocarcinoma and the like. So that is another key thing. So I really think that in parallel, there has to be a lot of education. There's a lot of support and engagement with the community oncologists to get them on board with this, because otherwise, it's still, we're going to be at the, we're going to be in the facet of science being way ahead of logistical implementation and clinical application of this. And that's a huge divide that really is important to bridge. And finally, I just wanted to make other comment, which actually goes it's a slightly separate comment, but it kind of tag teams with a couple of comments by Dr. Pantella as well as Sevanthi earlier, where there was some discussion about having to do many more clinical intervention trials to determine the clinical utility, especially when it comes to monitoring with serial measurements of CTCs in the metastatic realm of how you monitor these patients, and utilizing the data to actually make strategic decisions about when to pivot patients to different therapies as they develop resistance. So, one thing that I think about a lot is, how is this going to impact the current guidelines, the NCCN guidelines? Because if you think about it, it's really static. And once you implement a guideline for a couple of years, it stays there. It doesn't really change. And now, this is a whole other ball game that we're talking about. I think we don't even understand what the implications truly will be, but they will be of huge and extraordinary significance in how we practice clinical oncology, because it's going to be dynamic. One. The second thing is, now you're talking about personalizing it to various patients. So let's say, for example, a patient through he has metastatic cancer, and through serial assays determined to have progressive mutations due to resistance mechanisms. So that means that he has to be on a specific regimens which are kind of discordant with the NCCN guidelines. So where does that leave us? Because at the end of the day, these have practical implications. Does the oncologist feel comfortable in making that decision all by himself? If the patient has an untoward outcome, who's going to explain to him that, what is the basis for him to, reassure the patients that he did it in good faith and this was standard, that this is a standard mechanism? And, what are the legal implications of that? So, that really is in a nutshell my perspective from much of what has been discussed over the past few hours, as well as the questions that you put forward there. I hope I was clear, and I hope that it provided you with more food for thought.
Thanks a lot. And I think we will skip the questions, because what I could see that the time is getting in India 11:38. And if we will further extend, we are going to change the date. Okay, so let's complete the discussion. I thank you all, the panelists, for your time, and I really appreciate your comments. And I will just now give this to Sevanthi and Dr. Mani. Please conclude the meeting, please.
So I'm just going to stop sharing. Prashant, we didn't ask the most controversial question.
Oh my God.
We had a fight about this before getting
I was thinking the same thing. For what reason I found? I'm just thinking same thing.
Can we do chemosensitivity on CTCs? And
yes.
What was the other thing? So,
that is for your next meeting. Prashant will have data of our work soon.
Oh my God. So, thank you, Dr. Govindababu.
Yeah, we have to start doing chemosensitivity on CTCs, and we have to report that. And what about PDL1 on CTCs and all of that? And I think, if we open that, it's a Pandora's box. But, we shouldn't have missed the opportunity to ask the thought leaders. We have, though. So I give it over to Dr. Mani Sendurai Mani for thanking the guest speakers. Mani, are you there?
Yes, I am here. I'm here. So, again, my sincere, I shouldn't say my alone, it's our sincere thanks to you all for staying up late. Rick, I feel I'm so sorry, Rick, for making you stay awake this late while tomorrow your sailing is waiting for you. I'm sure you will be fine on the sailboat. And I thank all the rest of you, the midnighters right now. I think as in another 30 minutes, we would call the third day symposium, 20 minutes. And I thank you all from our hearts. It's not an easy task to, again, many times we thought that people may come, give a talk and then run out, but it's not. You all stayed up and stayed late and participated in the discussion. And that's the key. Again, I always dream, one of the things which is missing in India, for example, in general, because I'm a graduate of Indian Institute of Science, Bangalore, I always dreamt that the clinicians and the scientists talk to one another. The more we do, the more, the people, the public can benefit. And this forum literally did that, right? And we got a perspective from a clinician. What do they want? What is really necessary in order for us to make a difference to the patient? That's our goal at the end of the day. And we got a perspective from a scientist to see what we are thinking and how can we solve the problem which is put forth in front of us. I think together we can really solve a number of problems. Particularly, this is the key problem. The ctDNA really transformed the way we are treating patients today. And really, I thank every one of you who stayed up late. and also the participants, the audience whom we don't see all of you, but we thank you all for putting questions here in the comment box, and then participating not only as an audience but also in the discussion. We will try to put together many such symposiums to have mainly interacting the two side of the equation. And we thank you all for everyone staying up late, particularly the panelists again, Andreas, Prashant, for moderating the session. It's really wonderful. It's tough to moderate, particularly when people are talking and just stopping them and stop. It's difficult, but thank you. And Dr. Kashi, we heard a lot about you, and I'm glad to meet you. And of course, Sevanthi has done a spectacular work. She's the network, the glue, who is kind of connecting all the dots, and I really appreciate and I enjoy working with her. Dr. Govind Babu and Rick, of course, thank you. I'm just going around the screen in which I see the name. So Rick for staying up, and Dr. Raja again for all your insights, and Hima for your input, and the rest, everyone who's on the panel, which I don't see any more of their names here. So thank you all. And with that, we wish you the very best, and hopefully the next time we will meet in person. As Klaus said, we will meet for a glass of wine. Yes. And you all have a wonderful day, and good night, good day, good morning, good evening, all that.
I'll just take 1 minute, just 1 minute. Prashant, we'll just do a 30-second thank you to all the supports. And for that, can I please see the slides? So majorly, this is our student team, Dr. Chetan, Aditya from IT, Dr. Ruturaj, and Janni, which we heard Janni's name throughout. We really, but Janni was the front, and these three guys working in the background really to keep this seamless. And next slide. And the team that is hosting us, really, truly a huge help from Intas Oncology to help us. Akhilesh, who I reached out to initially to help me. There were just 2 weeks, not even 2 weeks. I think Akhilesh I reached out when there were 5 days. Ashish, really keeping it seamless. Anis and Amit Sethi. Thank you so much for all your help, and bring it back to Prashant. This was your show. And so I'll let you conclude. Thank you.
So, yeah, thank you all. I think the goal was that we will start this virtual meeting with an effort to bring the pioneers, experts, clinicians, and industrialists to address the challenges of early detection of cancer and the potential of liquid biopsy. And I think we somehow managed to justify it. I again thank you from the bottom of my heart, all the participants and the speakers, and thank you very much for all of your effort. And I think Sevanthi has done a spectacular, as what Bunny told. So without her support, I wouldn't have stand here. And Mani, thank you very much. You were the person who was supporting us from the back, but trust me, your support pushed us here, and we are standing. So thank you, thanks a lot. And yeah, thank you very much. Stay safe.
One
Good
last
night.
thing, Prashant. One last thing. Thanks to Kokilaben Hospital. Thanks to Datar Cancer Genetics. Thanks to Foundation One. Thanks to Institute of Bioinformatics. Thanks to Ilon Oncology. All of these people who have supported us in bringing this meeting together. Thank you all.
Thank you all. Stay safe. Good night.
Thank you. Good night. Fantastic work. Well done.
Thank you.
Thanks, everyone. Good night.
Bye.
Good night.